All right. You all are the privileged ones who actually showed up on time, so you get the full experience, and those laggards will have to miss this amazing intro. But welcome, everybody. You guys are here for the advanced tutorial on clinical characterization. Hope you're all in the right place. We're going to have a lot of fun this afternoon working together. I'm Patrick Ryan. I am at Johnson & Johnson and Columbia University. And I will be doing the minor part of this tutorial, which is just to orient what we're talking about when we say clinical characterization. And then we're going to get a lot of great advanced tutorial sessions from our all-star faculty. You've got Marty, Azza, Aniek, and Cindy, who will all introduce themselves as we go through our respective sessions here. And you've really got some of the folks that have been actually leading the work in the OHDSI community to develop characterization tools that are faculty to help you all along this journey. What we are actually going to cover today, I'm going to give you a high-level overview of the characterization use case as we think about it in the OHDSI community. From there, Marty is actually going to do a drill down into the DARWIN-EU tools. DARWIN-EU is a large EMA project that has a very large European data network, and as part of that, Marty has led the development of a wide collection of tools, open source tools that you can take advantage of that all run against the OMOP Common Data Model. So we're really lucky to have Marty here. After that, Azza is actually going to do an interactive exercise. So you need to stay awake because what I'm going to present and what Marty's going to present, Azza is going to actually then challenge you all to think about how you could use the tools we show you in designing your own studies. From there, Aniek is going to do a deeper dive into treatment patterns, which is a OHDSI tool that also came from our DARWIN-EU collaborators that Aniek originally started as research and turned into one of our production tools, and is a fabulous tool for enabling us to look at drug utilization. So you're going to get lots of details there. We'll take a break if you're good. And then we will come back, and Cindy's going to do a deeper dive into estimating incidence rates. Then Aniek's going to talk about risk factor analysis, which is very oftentimes a thing that people think they would do in a prediction class, not a characterization class. But Aniek's going to explain to you why you're in the right place in characterization to learn about that. From there, Azza's going to have another exercise for you. You're going to get a chance to play with some of the OHDSI open source tools for characterization. And then I'll close it up to talk about how hopefully as a next step, this class motivates you all to want to design and execute your own OHDSI network study. Sound good? All right. I saw 2 head nods, so I'm going to take that as enthusiasm. So first, I'm going to walk through our overview of the characterization use case in the OHDSI analytic ecosystem. So in OHDSI, we generally have been classifying that there are 3 primary use cases for real-world data. There is clinical characterization, which is what happened to them. We can think of this as descriptive epidemiology from a wide array of different use cases, whether that be disease, natural history, outcome incidence, quality improvement, treatment utilization. We also do a lot in the OHDSI community for patient-level prediction, so the application of machine learning algorithms to try to predict future outcomes from patients. So this is non-causal inference. And if that's a topic that's of interest to you, there is another wonderful tutorial that's being led by Jenna just down the hall. But you're still in the right place here for characterization. And then our third use case is population-level effect estimation, or causal inference. And so here we're actually trying to explicitly be clear about how we would generate a causal effect. And there's another tutorial going on right now led by George Ripsack that's on that topic. Now, the main thing I want to reinforce is that while you're here in the characterization class, it's not that we have any value judgment about these use cases. We actually think of them as complementary. And so I would say whether your goal is to do a characterization study as the endpoint, of which all of us as faculty have published numerous papers just doing characterization, and that's a totally valid use case on its own, or you want to do a prediction use case, you still need to do characterization analysis. Or you want to do causal inference, you still need to do characterization analysis. So no matter what you want to do to learn from your real world data, it's probably going to at least start, if not end, with characterization, which is why today we're going to focus on that top box there. Now within OHDSI, we have, you hear a lot about the discussion of how we standardize data to the OMOP Common Data Model, and we've standardized analytics through a series of open source software tools. But one of the things that we've actually recognized is that in order to develop a way to apply standardized analyses to standardized tools, we actually need to start by standardizing the questions that we ask. And so there are a variety of different questions that one could ask that fall into these categories of clinical characterization, population level effect estimation, and patient level prediction. For this tutorial, we're going to focus here in on characterization. We'll talk about disease natural history, treatment utilization, and outcome incidence as examples. But the idea is that no matter what disease area you're interested in, no matter what medical intervention you're interested in, whether you're interested in drugs or procedures or measurements, there's probably a very standard rubric for how you can think about asking questions. And a large part of this tutorial is going to help you understand how to structure your questions such that you can then, once you know the question and have an expectation of what the output could be, then you can think about using standardized analytics on standardized data to generate that evidence that you're looking for. So as a specific example, imagine that I was interested in a basic clinical question, like I just want to study ACE inhibitors and their effect on some outcome like myocardial infarction and angioedema. We recognize that ACE inhibitors are a drug for hypertension and that ACE inhibitors are one of many alternative treatments for hypertension alongside other comparator drugs such as ARBs. So if I just had this broad context, I'm a researcher and I want to study ACE inhibitors, well, I can actually narrow that down to say, like, well, how would I specify a series of clinical characterization questions? I could be explicit about the fact that I want to do disease natural history, which probably means I need to understand who are the people who have hypertension. Or I need to be able to specifically characterize who are the people that are taking ACE inhibitors after they've had hypertension. I may be interested in treatment utilization, and explicitly I'd be asking a question, amongst people with hypertension, what's the series of hypertensive treatments that those patients have taken? Or I may be interested in outcome incidence, where I'm saying amongst people who are new users of ACE inhibitors, how often do we actually observe acute myocardial infarctions occurring in some period of time following exposure? All of these are just kind of standardized questions where you can fill in the blanks to these types of questions, and across the characterization, prediction, and estimation use case, if you're able to kind of fill in these blanks, you can collectively create the set of evidence that may be of interest to you. So we're going to focus here on characterization, and to level set a bit further, I'm going to just use this caricature of a patient journey to kind of motivate what data can look like for a given individual. So here in this cartoon, I'm just representing a single person's cartoon data. On the x-axis, I have time. On the y-axis, I'm just showing you the different dimensions of data that we commonly think about. You can think about these like the domains in our OMUP common data model: conditions, drugs, procedures, measurements. And for any given patient, a patient is going to have a series of time-stamped observations scattered across these different domains. And very commonly what we want to do in any characterization analysis is partition that person's longitudinal record into some explicit intervals of time. For example, if I'm interested in that ACE inhibitor use case, I may say, well, I'll index on treatment. And once I index on some specific observation, now I can ask questions like, for example, like what diseases do I observe prior to treatment? What outcomes do I observe after treatment? And so being able to take all these longitudinal time-stamped observations, identify some sort of index date, now allows for you to define an analysis in a series of time windows to study things both prior to and after exposure. I see a few of you taking pictures of slides. Totally fine, or maybe you're taking a picture of me because obviously. But just so you know, all of the slides that we're making are going to be posted on the Odyssey website on the tutorials page after we're done. And I'll also, since I emailed all of you, I'll also just send you the link so that you have it. So don't feel obligated, but feel free to take pictures of me if that's your thing. Okay, so we've got our patient profile here. What are the types of questions we could ask? So when I say disease natural history, you could think, for example, what are the characteristics to a patient with a disease? What characteristics do they actually have? Or when I say treatment utilization, it may be things like how many patients with the disease actually get treated? Or which treatments do a patient choose after they're diagnosed? And when I use a use case like outcome incidence, we could say, how often does an outcome happen after treatment starts? We can also think about a characterization analysis as a time to event. So for example, like when do outcomes happen relative to exposure? All of these are standard characterization questions that just involve producing descriptive statistics to summarize a given population. And so what I'm going to do in the next 15 minutes or so is just give you a very quick whirlwind tour through these different use cases, showcase to you what tools are available in the OHDSI community, and then for the rest of this afternoon, you're actually going to get that deeper dive from our faculty to go through each of these individual tools. So I'll start here with disease natural history. So you could think of the default template, fill in the blank here, as like amongst patients who are diagnosed with some sort of disease, what are the patient's characteristics from their medical history? So here you could index on a particular diagnosis, for example, and explicitly say, now that I know a moment in a person's record, what's going on with those people? Who are they? What are their demographics? What are their comorbidities? What are their concomitant medications? All of these are the kinds of questions that we can answer in a standardized way. Now, the way that we often specify a disease natural history question is to try to standardize what are those inputs. And in this context, what we really need to do is define that target population. Who are the people that you want to describe? We can actually implement that very explicitly as a cohort in our OHDSI tools. And for those of you who are interested in phenotyping, there is a vocabulary phenotyping tutorial going on right now. We've got a very exciting phenotype work group activity that's happening on Thursday. Outside of the scope of this tutorial is just the idea that you can phenotype to create some sort of algorithm that identifies people who satisfy some criteria for some duration of time. Given that you have one of those phenotypes, it can serve as an input to these analyses. So in this context, what I'm saying is all I really need to know is what is my target population? That could be defined as a cohort. And that's basically just like, who do you want to do the characterization for? You also have the features. So what are the attributes about people that you actually want to summarize? And then for many different features, you will also have a dimension of a time window. When do the characteristics actually need to occur in relation to that cohort? Now, one of the tools that's available in the OHDSI community is a tool called Characterization. It is not a deceptive name. It is a tool that does characterization. And so if you wanted to run a characterization analysis, you could use the characterization package, and it would allow for you to, for example, pass in a target cohort, define the features, execute an analysis to get information about disease natural history. Now, in the OHDSI community, we've done quite a lot of disease natural history studies as a community. I'll highlight some papers just to kind of help you motivate thinking of like, well, what kind of questions would I potentially want to answer? One of the large characterization studies we did during the COVID pandemic was initially just trying to understand what is COVID looking like. Early on, you may remember that there was a lot of questions about like, what is COVID? How does it compare when you're already been hospitalized for COVID versus you've been diagnosed, but you haven't yet been hospitalized versus you've tested positive, but you haven't yet been identified any symptoms. And as a community, we conducted a large-scale characterization study called Characterization and published a large collection of different characterization results. The motivation for this was we need to understand the disease before we can even think about going on to studying it, whether that be to find effective treatments, to prevent the event. First, you just need to understand what are you actually looking at. And in this particular context, one of the most important things we can do in the OHDSI community is run network studies around the world. Because disease natural history for COVID looked a lot different in China and Wuhan at the start than it did in Korea, than it did when it moved into Italy, than it did throughout the rest of Europe, and than it did in the United States, or even regions in the United States. And so disease natural history as a network study provides a tremendous opportunity to understand to what extent are there similarities or differences in how diseases present in different databases. Treatment utilization is another compelling use case. Here you can think of like amongst people who have some sort of diagnosis you're interested in, which treatments were patients exposed to and potentially in which sequence. So for example, if I'm saying, amongst patients who who go on to have a heart attack or maybe have hypertension. We say, like, what hypertensive drugs have they had in what order? Here, in order to actually specify this, you need to define your target population of interest. So that could be like your people with hypertension. And you also need to be able to define all of your events. In this case, the events would be all those potential medical interventions you're interested in studying. You also have the ability to think about when doing a treatment utilization, it's very frequent that we're not just interested in looking at the target population as a whole, but we may want to purposely stratify that target population into subgroups of interest. Most commonly, and in fact, as a scientific best practice, we encourage thinking about stratifying on things like age and sex as a standard way to make sure that you're seeing consistency, as well as for treatment utilization. Most commonly, we almost always want to stratify on time because we know treatment trends change over time. Now, one of the OHDSI packages that you can learn about is treatment patterns. This is the tool that Aniek's going to do a deeper dive into, and it provides a wonderful mechanism for you to be able to characterize these pathways with large-scale analysis and really nice visualizations. Just to provide an example of like how this has worked in the OHDSI community, one of our first OHDSI network studies was actually characterizing treatment pathways, and we did this analysis explicitly just to think about the extent to which we could run global analyses and whether or not we could gain any particular insights. And in this particular case, what we did is we actually asked the question, like, how are patients with depression actually treated? And this sunburst representation, what this is showing you is the inner circle is basically the first line of treatment. The outer circle is the second line of treatment. The third circle is the third line of treatment, so on and so forth. The colors represent different drugs. In this particular case, when we ask the question, how are patients with major depressive disorder actually treated, we see tremendous heterogeneity in the patient experience. Even as a first-line treatment, you can see that there's 10 different drugs that are very regularly commonly used as first-line treatment. And then once one of those fails, there's another 10 drugs that patients are seemingly almost like randomly being assigned. And you can imagine that it's very difficult to manage a disease if you've got all these treatment options and there doesn't seem to be any specific guidance towards one particular treatment being effective as a first-line therapy. Now, in that first study, we actually we looked at depressive disorder across the network, but we were also able to look at that within each database in the network. And actually all of these sunbursts actually represent the different treatment patterns we observed in the different databases. And what was actually particularly revealing and exciting was that for one of the first times, we're able to start to learn about heterogeneity across populations by doing this type of analysis. And so it's one thing to do a treatment pattern analysis, say like, what's going on in my database? But it's another thing entirely if you can benchmark what did I see in my population to what would other people see in other geographies or data capture systems. And this study actually was tremendously useful for us in understanding the value of heterogeneity across the OHDSI community and how we could use that as a real catalyst for the type of research we do. But a treatment patterns study, while it can be an endpoint, and we have seen numerous publications from our community just stopping at a treatment patterns, very often what the most exciting thing about a treatment pattern study isn't the question that it answers, but it's the questions that it prompts you to ask. So for example, if I see one of these patterns, I can ask, which treatment did patients choose? That's what the treatment pattern actually shows me. But I could actually start to think, well, which patients chose which treatments? Or how many patients who went down this particular treatment pattern actually experienced an outcome? Or what's the likelihood that I'd actually experience an outcome given that I had this treatment sequence? Or does the treatment actually cause the outcome is something that we can first see, well, how many people do we actually have on a particular line of therapy to think about whether that path is actually safe and effective? And so I think while Aniek and Martin and the team have made a tremendous package and we're starting to use it, I think it's a critically underutilized resource in our community because of how it can tell us so much about data to prompt the kind of future research that you could do. Another use case we have is incidence rates. So you can think of like amongst new users of a treatment, how often do they go on to experience some outcome within some time horizon? To define an incidence rate analysis, you basically need to specify your target population. So that's your cohort of people at risk. You need to specify your outcome. So those are like the events you're trying to actually estimate the risk of. You'll define that time at risk period to be that interval when the outcomes can happen amongst the time at the target population. And much like that treatment patterns analysis, you may think that you want to study specific subpopulations. And explicitly in our prior research, we advise all incidence analysis should be stratified by age and sex. Now, we have multiple packages in the OHDSI community. The one I'm highlighting here is Cohort Incidence, which is a tool explicitly designed to allow you to take a target cohort and an outcome to conduct analysis. And we've been using this tool extensively across the OHDSI community for lots of studies. Marty's also going to highlight the incidence prevalence package that the DARWIN-EU team did that does a complementary type of work. Just to motivate the example here, incidence rates. During the COVID pandemic, one of the questions that regulators were asking was, what is the background incidence rates of adverse events of interest, such that as vaccines started to be used, we had some sort of frame of reference to benchmark against adverse events that were being observed amongst those exposed to vaccines. And so our paper we had a series of papers, one published here in BMJ, where we actually provided the network estimates of incidence stratified by age and sex across the entire panel of adverse events. And this information was actually used by the European Medicines Agency to inform their vaccine regulatory policy. I'll highlight also time to event analysis. This is actually a use case that we hear people talk about, usually in the context of either prediction or characterization, but I'll assert to you that this is a bread and butter characterization type analysis. The basic idea is that you have a target population, those that may go on to have an outcome, and you have an outcome, those people who are the actual events. Our characterization package actually offers the ability to provide you a descriptive summary of the time to event relationship between the exposure and the outcome. Now, why would you potentially want this? I'll motivate it by going back to our discussion of ACE inhibitors and thinking about two different use cases. So ACE inhibitors are a treatment for hypertension. You'd usually take that drug to try to reduce the risk of myocardial infarction. However, ACE inhibitors are known to have a side effect, which is angioedema. And so just to motivate this, we might be interested in looking at a time to event plot. This is a time to event plot. Let me just orient you to the graph. On the x-axis, we have time relative to time zero. That's your index date. That's when you entered the target cohort. And then on the y-axis, it's a histogram, so I'm just showing you the number of events. So you can think of this as how many people took the target and had the outcome. The histogram is now plotted, providing you the frequency of occurrences relative to time. And the colors are telling you specific dynamics about what kind of outcome and what kind of exposure you're looking at. In this particular case, for myocardial infarction, we see a really interesting pattern that indeed people who are treated for hypertension might have heart attacks. So the whole reason you're treated hypertension is to reduce the risk, but it's not going to eliminate the risk. But what's really interesting is actually this orange bar up here. We can actually see that what's a big motivator for managing your hypertension? Having had a heart attack. If you have a heart attack, then they're going to be more serious about managing your disease, including your hypertension. And that, in fact, this is showing lots of people who have ACE inhibitors and heart attacks actually have the heart attack first, not second. That's actually in tremendously important context, particularly if you're going to study the safety and effectiveness of ACE inhibitors. It's probably important to know, why were you starting on the ACE inhibitor in the first place? It might have been because you had a heart attack. And so we can find this to be useful both to understand when events happen, but also like what happened even before you were exposed. Now, in contrast, I mentioned that ACE inhibitors increase the risk of angioedema. This is a rare side effect associated with swelling, and it's known to be related to ACE inhibitors. It's one of the things that they watch out for. In this particular context, the same graph, what you can see is that we don't see very much angioedema happen prior to exposure, but then all of a sudden, within the first 30 days of exposure, we see this tremendous leap, and we see quite a lot of angioedema events happening. And this can be a really important characterization analysis, both just to be able to say, like, I'm interested in studying whether or not there's a causal relationship between exposure, ACE inhibitor, and the outcome of angioedema. I'm going to do causal analysis. I'm going to go learn from George. But before I actually do that, I might just want to know, like, well, where are the events? When are they happening? And what do they look like? And this can actually help to inform even the design of future causal inference questions. one more minute, 2 more minutes, that I'll summarize. Risk factors is yet another use case that we're going to talk about, and actually Aniek's going to dive in deeper to this. The argument that I'm going to make here is that very, very frequently we hear people talking about wanting to do prediction studies to learn risk factors. But effectively, the question of a risk factor is amongst some sort of population, you could have some sort of referent comparator population and some sort of time at risk. And you'd ask the question, amongst people who go on to have the outcome versus those who do not go on to have the outcome, what are the differences in those baseline characteristics? And lo and behold, when you do that, you actually are just explicitly learning a risk factor without doing any fancy statistical modeling. So to use this example, if we ask the question, amongst those people who were exposed to ACE inhibitors who went on to have a heart attack, as compared to people who had ACE inhibitors and did not go on to have a heart attack, what are their differences? And we can do a descriptive characterization to learn that indeed, the people that are more likely to have a heart attack are the people that already had heart disease. The people who are more likely to have a heart attack are those that have COPD. The people who are more likely to have a heart attack are those that are smokers. Now, those are risk factors, but we don't need to do any fancy statistics to actually learn that. And in fact, Aniek's going to show that if you do fancy statistics to learn that, you can actually be misled. That characterization is actually the right and appropriate way to get these answers. So with that, I want to just summarize to say, in characterization, there's a lot of questions you can ask. They can either be the questions that is the question you actually want the answer to, or it could be the thing you should learn before you go on to answering the question that you want to ask. In either context, whether you are interested in disease natural history, treatment utilization, outcome incidence, time to event, risk factor analysis, in the OHDSI community, we have a whole suite of OHDSI standardized tools. We're going to give you the opportunity to think about how to design your questions using this standardized rubric, and then help you think about if you've already come up with that right question, how do we actually help you find the right tool to answer that question? But before we have you do that exercise, right now I'm going to turn it over to Marty, who's going to go through even another whole batch of exciting characterization tools that are available within our OHDSI community developed by the DARWIN-EU team. So with that, I'll call up Marty and I'll flip over the slides. Oh, and I totally failed. I'm sorry. We want to encourage participation. I just blathered at you for 30 minutes and didn't say that. That's awful. I'm sorry. I'm the worst. To encourage characterization, encourage participation and characterization, both of those things at the same time. We have a lot of candy up at the front of the room. Anybody who asks a question or makes a comment, they get to pick their own box of candy. So does anybody have any questions or comments? Rima.
What if the population and target are the same, or if we just have a population that don't have a target? What do you do? For example, surgery waiting time. Surgery is an outcome. Population is cancer. Different type of cancer. There is no target.
Yes, when I say the word target, what I just mean like of interest. So it's your population of interest. So your population could be people who got a surgery, people who have a cancer. We need to be able to define a time point associated with that because you're going to want an index date. So it would be people who had a cancer at the time that they were diagnosed, people who had a surgery at the time that they had the procedure, that type of thing. It is your population of interest, and we just refer to that as your target population. And Rima, you get to pick. I went to Dollar Tree, and I literally bought one of every candy they had so that you incentivize asking questions quickly, because then you get the best pick of the candy. So Rima now gets the very first favorite choice. Anybody else have any questions or comments, or just want to get candy? Peter wants candy and has a good question. Peter, you won. You get a second box of candy. Well done. He played me.
Good afternoon, everyone. I'm Martí Català. I'm a senior data scientist at the University of Oxford. And well, I'm here because I'm highly involved in the DARWIN-EU development. And I'll try to explain some complementary packages and what we do in the DARWIN-EU. First of all, Oops. Disclosure that basically whatever I say only concerns me. And yeah, what I'm going to explain today. So first, I don't know if everyone is familiar with the what's DARWIN-EU project. So I'm going to give a brief introduction of what's the DARWIN-EU initiative and how it's managed and how it works. You have the director there, so yeah. Then I'm gonna focus more on the characterization pipelines in the DARWIN-EU. And finally, with the time that I have, I'll do a hands-on practical, showing how we would run some code, so you can get an idea of what I explained, how it's translated into code and into the practical. So yeah, first of all, DARWIN-EU. What's DARWIN-EU? So DARWIN-EU is a big project funded by the European Medicines Agency and the European Medicine Regulatory Network that basically they created, they set up a coordination center that it's responsible for have a network of data partners that and then provide support for regulatory decision making. Basically, they ask questions, we give answers. That would be the summary of what's Darwin. And we do this within with a big network. Now we have 40 data partners. And if I'm speaking here today, is because everyone is mapped to the OMOP common data model. So we do it in a federated, what we call federated analytics. That basically what means it's EMA has their own structure. No. Can I put the cursor somewhere? Yeah. So the EMA has their own structure and committees that usually are the ones that come with the questions that they want to be answered. And they give these questions to the coordination center in the EMA. The EMA go to the coordination center in DARWIN-EU. That is the one responsible from creating, okay, you want to answer this question, so this is how we are going to answer. We create a protocol. These are, from these 40 data sources that we have available, we are going to use 5, 6, 7 that are these ones. These are the ones that seem to have the conditions and so that make more likely to produce the answer that to the question that you want. Blah, 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 prepares a code, and then this code is sent to the data partners. Data partners has their data in-house, so we don't have access to any data. The coordination center is only responsible for overviewing everything. And then the data partner has access to the OMOP common data model, runs the code, the results are aggregated as statistics, that then they go back to the coordination center. The coordination center uses the results from all the different data partners to build a report. This report is sent to the EMA, that the EMA informs the scientific committee of interest. And this is how Darwin works. And okay, what do we do apart? I said answering questions, which is a very broad thing to do. I answer questions in many things. So what do? We have the catalog of standard analytics that basically we have tabulated that the studies can be of 4 different types. I'm going to focus only on the first one today, but I want to explain the context of what we've done. So we have the off-the-shelf studies that are I was going to say simple, but probably if some of the data scientists hear this word, they're going to complain. But simple studies where everything is more tabulated. What they can answer, it's tabulated. Then we have the complex studies that it's all the opposite. It's answer whatever you want, and they can be more complex. And then we have 2 extra types that routine repeated is basically a repetition of a previous study that we've conducted in previous years. And very complex is requiring usually adding something more apart from the complexity, adding map data of genetics, adding things that have not been done, or mapping extra concepts, more complicated stuff. We are just doing the first 2 very complex studies. So today I'm going to focus on these off-the-shelf studies, and then what do we do in these off-the-shelf studies? As I was saying, it's like studies that they've been tabulated, and what you can answer, what you can ask, it's limited, and you can ask for 4 things. 1, patient-level characterization, like characterize a set of patients. I want the characteristics of patients who have dementia. I want the characteristics of patients who had venous thromboembolism, whatever. We do like patient-level drug utilization. That is like the same, but at the drug level, like prescriptions, duration, all this. Population-level drug utilization, that is like use of drugs, and population-level descriptive epidemiology, that is incidence and prevalence and so on. So to answer this, what we've done is to build R packages that match the different questions. So to answer the patient-level characterization, we have cohort characteristics, we have cohort survival if we want to deal with survival, cohort characteristics to deal with the different characteristics of the population, and then with the drug utilization analysis, we have drug utilization package, the treatment patterns package, and so on. And incidence prevalence is the one used to do the population level studies. So basically, we've developed all these packages, and what we've done is not only to develop these 5 packages and what I'm gonna talk today, it's patient-level characterization. Just like, okay, I've explained the whole story to end up here, but like just that we know the context of what I'm gonna explain. And just to mention that, well, to support all this work is not only those 5 packages that I showed. So we've built a whole infrastructure of 12 packages that work together also with other Odyssey tools. They are open source, they are available in CRAN, they are in GitHub, so everyone can access them. Each one of the packages has its own website, documentation, and everything. And we are quite happy that we've reached the 200,000 cumulative downloads like in the last 3 years that this existed. So this is the context, and 8 minutes is fine. So characterization, now I'm gonna focus on the characterization pipeline. If we look at if you remember before, I said, I'm gonna talk about this, patient level characterization. So if you look, what we promise in the patient level characterization is large scale characterization, patient level, large scale characterization. I'm gonna enter in the details later. So large scale characterization, patient level characterization, pre-specified patient level characterization, incidence rates, prognosis, and like descriptions and so. So then, okay, we can do all this. And then Patrick told me, you have 30 minutes. And then it was like, okay. So what I'm gonna do is to focus on the ones that are like involved. And in particular, I'm gonna explain from those 12 packages that I said that exist in one of the packages that it's cohort characteristics. And then what cohort characteristics can do. Okay, so cohort characteristics covers 5 things of the catalog of standard analytics. One, it's to visualize and extract the attrition. Like here, we don't do any magic or anything. We just do a plot of the attrition. The other 4 are actual analysis, where what we do is like visualize overlap between cohorts. And you can do this overlap at the person level or also at the index date level. Then to visualize timing between different cohorts. This is quite interesting when you want to see if some events come before the other and so on. Finally, and the most important one, usually what we call like in epidemiology, table 1, we can build table 1 to describe the demographic characteristics, cohort intersections, and all the characteristics of like a certain target population. And finally, the large scale characteristics. It's a data-driven thing. So we are gonna go through the different stuff. So first of all, to visualize and extract the attrition, what the package produces is just a simple diagram where we can track the different decisions that we've taken along our cohort definition. This is quite important to later know which was the impact of our decisions and so. So as you can see, if someone would see this and would have seen like the Atlas, if someone is familiarized with the with Atlas tool and like the Atlas attrition, this is a bit different, but it's more like at the step-by-step level. The Atlas one provides like more intersections, but yeah, it's just to mention it's a bit different. Then second thing that we can do is to the overlap between cohorts. There is basically, then we would classify, we would have like two cohorts. In this case, it's like a study about antipsychotics and dementia. We wanted to see which was the overlap between people taking antipsychotics and taking dementia. Basically what we saw is that there were, most of the people were only on the reference cohort, it's antipsychotics, so there were more people. There was an overlap, and this depended like on the different databases. We saw different patterns. But yeah, that's what you can see, and like you can analyze what's the overlap between different cohorts. Finally, I'm going to more interesting stuff. In the same study, what we did is like to visualize the time in between cohorts, and then we did the same, instead with dementia, insomnia. But to see in individuals who have both the record of antipsychotics and insomnia, so it would be in this plot, taking only the green patients that are the ones that are in both cohorts, we analyze the timing or what's the event that is coming first. And in most of the studies, I was not explaining it correctly. So it's intersection between dementia and antipsychotics and dementia and insomnia. That's why, yeah. So it's exactly what I was showing before. In this, using the green patients, and then we see that in general, dementia and insomnia were like there was not a clear trend. Like maybe insomnia was coming a bit before, but not much. But we could see a clear trend that like antipsychotics were starting after dementia in most of the databases. Here, for the ones that are used to work with multiple databases, there is always one that like points the other way around. In this case, NICE, that is a Croatian database, like was showing different results. But yeah, that's live and real world data. And yeah, so, okay, since the moment, I don't think I showed like anything spectacular, but that's things that we can do with the package. And now the main purpose of core characteristics is to build like what we call like the table one. What we include usually in table one, first, demographics of patients at index date, like age, sex, typical stuff. Then things like what are the medications that the patients were taking, and then we have like, okay, taking when. Usually we do medications the year before, but we have the flexibility to define year before, month before, week before, only at index date or whatever like of interest depending on the study. So like this window is defined study by study basis depending on your question. The same for conditions. In the conditions case, usually we take like a longer window. We take like any condition that was diagnosed before, but again, on a study by study basis, sometimes like you are only interested like in conditions diagnosed the year before for some reasons. And usually we also include something related to health-seeking behavior, like how many visits the patients had like during the year before, just to have an idea on like if you have very sick patients or less compared usually when you are comparing two different cohorts. That's quite interesting. And just to show just very quickly a result, this is a result from like the opposite study that we conducted. It was a routine repeated from like year 2. So this is a year 3 study that was also conducted in year 2. And yeah, basically we had like 7 databases, and like I don't want to point out like any of the results, but you can see like how many records, how many users we had, how many subjects, like there could be like multiple records per person, age, we have median age, quantiles, the range, Then percentage of sex, for example, we have in one database, we have like some unknown sex, we have treatment duration, and we have also comorbidities anytime prior in this case. So yeah, this would be like the standard output. The nice thing is that these tables are produced in GT format. That basically what it means is that it can be exported to HTML, to a Word document. So you can put it like in the website, you can put it like in Shiny apps, we can put it in reports, or you can put it directly into Word. So it's quite nice to export output tables in this format. And finally, the last thing that we can do with cohort characteristics is to summarize the large scale characterization. I don't know if any of you is very familiar with this. I think Patrick mentioned a bit, but basically it's a concept level characterization of the different clinical tables. We have like an index date that it can be, depends on like our cohort definition. And then what we say is like, oh, what happened we define windows. What happened on the month before? What happened on the month after? What happened in the year before? So you define a window, and then you extract the percentage of individuals that have a certain condition, a certain concept ID for all the concept IDs. And yeah, we have the ability to also do it at the level of combining standard and source concepts. So if you know in the OMOP clinical tables, you always have like the standard concept ID and the source concept ID. So you can like only count grouping by standard concept ID or grouping by both of them. But yeah, just explaining this. So at the end, what you have is a very large table. This was like in HTML. And as you can see, there were like 11 pages. So it means that 110 like concepts were extracted, and then you have like the percentage for each one of them. And then you can compare the different windows, the different cohorts, depending on what's your question. You are gonna want to compare different cohorts, different windows, or different databases. Although this characterization is not very good to compare at database level, because each database might be like mapping at different levels. So, and this is always counts at the concept level. So maybe you would get like the wrong idea saying, oh, I don't know, there are only acute bronchitis in that database, but then there is another concept that is acute bronchitis and blah, 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 that then like the other database has counts on that one. So this is not good to compare in databases. But yeah, so you can do it. And similarly, you can also do plots. And in this case, like I just done a simple plot comparing male and females, and there are not much differences. But so like the diagonal, its reference, it was male, no. Well, there was two cohorts. I have to remember now what I was doing, but basically we were comparing two. So the closer it's into the diagonal, it means that the same value it was in the reference and in the comparator, and like the farther it's from the diagonal, it means that like it diverges more. And we stratified this analysis in this case for female and male. These plots are quite interesting, but always different to analyze. Each point is a different concept ID. But yeah, if you see a trend of like a cloud of points that like diverges from the diagonal, this means that you have like a seeker population, or like that there is something there to look at it. And yeah, I think I've managed in 20 minutes to finish what I wanted. So we have 10 minutes of the hands-on practical. Hands-on practical. Now, the EMA, we had like a very quick call, and they told me that they want like a new study. We have to conduct it in 10 minutes. And basically they want us to characterize patients at first sinusitis diagnostics between 1990 and 2009, stratified by sex and year of diagnosis, and they define 5-year bands, and to estimate the yearly incidence of sinusitis between 1990 and 2009, stratified by sex. So that could be a simplified version of how it would look like on a study. So it would be this but like adding lots and lots of text, like 50 pages, but that's the main message. So basically, we would have to conduct this. How we are gonna do? So now I'm gonna show how the code would look like. So basically, I've set up like a POSIT cloud that I'm gonna share later, so that anyone that wants to try, feel free to try. But yeah, basically, is it big? No. Is it fine? Okay. So yeah, basically I listed all the packages, just that if any, I'm gonna share the code and the POSIT cloud. So if any of you wants to try it at home, just to check that the guy who was standing there was not lying. So you can just install all the packages and check it out. But yeah, how it would work. So I load the libraries. Then in this case, I don't have any database, so I would download a synthetic one. So I don't think that the results are gonna be impressive or the EMA will be very interested, but like I'm gonna have some results. So I'm gonna use the GA bleed dataset. I don't know if any of you is familiarized with it. But yeah, how it works basically, we have to connect to the database, create the CDM object, that it's basically provide a connection and like say the schemas, a bit like the connection details that we are used to use with a database connector. But yeah, basically it's just a fancy print of like a reference of all the typical tables that we would have like in an OMOP database instance. Then, like just to show, it's like you would have, this is how it would look like CDM $person. If you connect to your own database, you would have like your own person table. Okay. Then the first thing, they said sinusitis, right? So I have to define sinusitis. One of the packages that I didn't mention, but it was there, it's code generator. It helps with this. Basically, it helps to do this in a standardized way, in terms that sometimes, otherwise, if you click a thing and you search sinusitis, something will pop up and like, well, it's basically the same idea. So yeah, I've searched sinusitis, 4 concepts pop out. We have 5 minutes. We are happy with this. And then I define a code list. So basically, I'm gonna use these 4 code lists that if you don't trust me, you would go to Athena, it should be the first. And basically, yeah, so it's this code of sinusitis. And then the first thing that I'm gonna do is I create my cohort of sinusitis, we run the query, and I've added like a couple of inclusion criteria. So I said, I'm only interested like in the first occurrence of sinusitis, and I'm only interested with events that happened between 1990 and 2009. This is because if you remember when the EMA, when we had the call with the EMA, they said, oh, we are interested in the characterization of the first sinusitis diagnostics between 1990 and 2009. So that's what I did here. And then basically we have our cohort that it's like a list. This is a patient level data. And so we've defined some metadata that can be useful. For example, now we know that we have 163 codes of individuals in our database. The attrition, I'm going to show it later, but it's part of the attributes. And you can still also like retrieve which was the code list that you used. Then the first thing that I showed, that I said that we would do is to summarize the cohort attrition. So I can summarize it and plot it. Now it should pop up here. Okay, so now I'm not gonna like deep dive like explaining it, but we would have like, okay, the different definition, the different decisions that we've made, that it was like, okay, record of sinusitis. Then we have some quality checks that usually it's good that you don't lose anyone here, but like you would have some quality checks to check that there is sex, that there is no like wrong dates and so. And finally, my decision to, well, the decision to only require the first entry trimmed like lots of records. It means that we have multiple people having like multiple records of sinusitis, and the same for the dates. We lost lots of people because we have lots of old records. This is like the toy database that we have. And then if we want to use the characterization, basically it just summarize characteristics and table characteristics. And it's quite nice because with just 2 lines of code, it took 10 seconds now. Like when you run it in your database, it's not gonna take 10 seconds, but you extract all this information that we wanted, like what we've seen before. It was just to show what I had like the slides. It was not me making it up. So it's like if you use our code, it works. So you get the age, you get the sex, you get the prior observation, future observation, how long they are in the cohort. So you extract all this information. And finally, because how we are doing on time, 5 minutes? All right. So yeah, and finally, what I'm gonna show is just if I add as columns. So here, what I've done. It just add one column that it's sex and one column that it's the year of diagnosis. And then what I can do is to say, okay, now I want you to, well, the package to stratify by sex and year of diagnosis. So before we didn't say anything. We said just summarize characteristics. And now I say, oh, in fact, do the summarization of the characteristics stratifying by year and by year of diagnosis and sex. And yeah, if I run it, it takes a bit longer, but also still fast. And I get the same information that I got before, but now I get like several groups. I have it for each one of the 5-year bands and for each one like male and female. So you would have like all that information to share with the EMA, or if you are like doing this, we also use package. I always refer to the EMA because Darwin is like EMA centric, but it's not that like these packages, if I'm explaining them now, is because they are open source and we use it like for many other studies, and I would encourage you to try them out. But yeah, you would have like all the same information. Now I've stratified by sex and by these 5-year bands. And finally, I'm not gonna go through it, but I'm just gonna run it very quickly. Believe that there is some code there, and then we could do something similar. It's a sequence of like running 4 queries, like first identifying outcome, then identifying the denominator, then calculating the incidence, and finally plotting it. And yeah, we would be able to have a plot of the incidence of sinusitis stratified by by sex through the different years. And yeah, so this has been like a brief overview of things that we can do with the Darwin tools. Now, my 2 last slides. The first one is this one. This is like a POSIT cloud that it's where I was running. It's just, the only thing is that it's an R instance that you don't need to install R. So you just open there and it's R in your browser, and it has the packages installed. So if you go there, this is an invitation. It's paid by Danny. So if you see Danny tomorrow, you can thank him. So at some point I will have to shut it down, but during the conference, it will be working. So if you go here, you will be able to run the code and modify it. In fact, I was proposing that if someone is able to change and instead of sinusitis, run it with pharyngitis, we can give like 2 candy boxes. So yeah, just encouraging people to try it out, just also like to see that what I've been explaining today for 30 minutes, like it works and how it works. And then finally, providing lots of links and things for you to take a look. So here there is the DARWIN-EU official website. You can go there, like all the details of the project and so. Then you have like the 4 analytical packages that I was mentioning. The first one, that is the one that I focused and I explained, it's cohort characteristics. Basically, if you would go there, well, probably I can show it when I finished. But yeah, you would have like a documentation with all the website documentation with everything that the package can do. And the same for incidence prevalence, cohort survival, or direct utilization that I didn't have time to cover. And also, I mentioned there are like 12 packages, so I also added like 2 other important ones. Then, not Darwin related, so that's what like I put it separately, but with my research group, we've built a book, we've written a book. It's online and freely available. It explains like some part like the packages and like how to set up stuff and so. Might be of interest, so if useful, I leave it there. And finally, you can try to send me emails. Yeah, I'm not the most responsive person, but I'll try. In GitHub, I'm way more responsive, so I also like leave my GitHub tag there, just in case. And yeah, thank you very much, and if there is any question.
Any questions or comments for Marty?
We have candies.
And candy, you still get candy. It's still eligible.
I'm going to donate my candy. Any overlap with Atlas or completely independent?
What?
This package, the DARWIN-EU package, and the Atlas, the similar Atlas.
Yeah, so like you can define a cohort in Atlas, instantiate it, and then characterize it with cohort characteristics. In fact, this is a quite common thing that we do. You use Atlas to define the cohort. I, for my background, prefer to define them programmatically, and that's what I did in the explanation, but it would work exactly the same if you do it like with Atlas, instantiate it, and then characterize it.
What was your incentive of doing it outside of Atlas?
So basically, probably this is about my pay grade, but as I take the question, so basically we needed like the control to own the code. We needed to be able like to fix bugs, to change things and so. So Darwin as a project needed to own the code, and that's what we've done. No. So this one leads to Posit Connect. This you can open, well, it's an R online account where you can like run code. It's installed, the packages are installed and you can run it, and yeah. And this barcode is like about a book. Could you open it up, please? That we've built.
I'll make sure before we're done today, all these slides will be posted.
Yeah, so you will have the, yeah, now this one is transparent, but yeah, sorry.
And you get candy.
I'm pretty familiar with the Cohort diagnostic package, and it seems quite similar but also different at the same time.
Mm-hmm.
Can I use the Cohort diagnostics rather than the utilization package instead?
So you can use whatever you want. Like you can, you will get complementary answers, and like each one of them has different stuff. So this, with cohort characteristics, you get, how to put it? So cohort diagnostics is more predefined. You have less control, but you will get like with not lots of code, lots of information. With cohort characteristics, you will have to make more effort in terms of you taking decisions of which medications, which conditions we have, but at the end, what you will get is more what you want. So I see them at different stages. Cohort characteristics is a package to do diagnostics. It's more, I've created a phenotype, I want to see if it works, if it generates the cohort that I want. So I instantiate the cohort and put it with cohort diagnostics. Cohort characteristics is more, I'm building the table 1 of my package and I want to control exactly what it has to go do. Yeah.
Now that you've heard from me and Marty, Azza's going to walk you through an exercise where you guys are going to be put to work. You should have gotten a handout. Raise your hand.
Hi, everyone. If you need to stand up, move a little bit, that's totally fine. We're going to do an exercise. First, my name is Azza. I am an OHDSI collaborator for now 7 years. I work with Patrick, and yeah, we're very excited to have you today. So what we're going to do is you all have a handout, and what we're going to do is we learned that we're talking about characterization. And one of the main principles of OHDSI is the whole idea of standardization. You have a standardized common data model, you have an amazing standardized vocabulary, but to be able to do a standardized research as well, we need to pose our clinical questions in a way that is we can then understand what's the intent, but it needs to be in a way, we need to present them in a way that is generalizable. So we can then learn how can we operationalize that research question and apply it on the data in a standardized format. To do that, Patrick, when we talked about characterization, on his slides, he presented a way that you could think of all possible clinical characterizations questions. And there were 3 buckets under that. That's what you see in front of your eyes right now on the handout. It's the 3 different questions that you can ask under the umbrella of characterization. And then there's an example of how you can pose that question. And from that, we can see how we can standardize the input from the question to be able to apply a standardized analysis. So now you're going to all put a hat of a researcher or an investigator where you have observational data and you have a question of interest that sits under the umbrella of clinical characterization. It can be about anything, not limited to any clinical domain. All what your task is to come up with a research question presented in the same way of that generalized format that we shared with you, and just write it down in that blank box. You can do this on your own, you can do it with your table, you can do it in pairs, whatever's comfortable with you. We're gonna just start writing that in your blank page, and then we're gonna start to understand based on your question, what would be those target population cohort, what's your outcome cohort based on the question. And then we're gonna discuss these questions along to each other so we see if we can learn, if we can truly represent all our characterization questions in this generic format, because if we do, we have a lot of gains in terms of standardization and applying the questions, letting the data answer the question in a way that is reproducible. Okay? Is it clear? Okay, think of a question on your own or with your friend, and I'll be coming around, and then we're gonna start writing your questions on that chart.
On patients with epilepsy, and certain drugs cause a very, very severe skin reaction. But I was just curious, even before that, like among pediatric patients, they're less than 18, who have had an epileptic seizure, what's the first drug that they're given? What's the sequence after that? Just curious. And then, like as I said, can you compare like one country versus another country, or private versus public, those kinds of things?
Thank you.
And then to simplify things, or to find all the drugs that are anti-epileptics, I know that there's an ATC classification. So if you put in like NO3A, it'll tell you all the drugs that are prescribed for epilepsy.
Yeah. Thank you. So we could generalize the question. Just see if we can generalize using the same format that Patrick had on his slide. So it's obviously a treatment pathway question. Do we all agree that we're interested in? Good. So we can say, and tell me if that describe your question accurately. So among pediatric patients less than 18 with epileptic seizure, which treatments were patients exposed to and in what sequence. And the drugs that we're interested in are all the ATC classification, so a long list of all those groups of epileptic drugs. Does that characterize the question? Okay, if this is the question, then what is our target population? What's the target cohort? So if we try to understand the question by saying there's a target population, there might be outcomes. So what do you think is the target population in this question? And to be more specific, pediatric patients with epileptic seizure. So that is the cohort of interest that we want to characterize. So that would be our target. Now we will need to build the definition and the phenotype to find this target population in the data. But now we know. What are the different events we're interested in among this population, target population? What are the different quote outcomes based on this treatment question? So we're going to characterize the children with epileptic seizure, and we're going to look at certain events that are happening to them because it's a treatment pathway, so we're looking at drug exposure. So that would be the different event. And because it's not one drug, so it's every ATC class that she wants to characterize will be an event cohort that we will need to define. And I know you're going to hear more about treatment pathway and how it's done and how it's standardized in the next section. So stay tuned for that. Okay, go ahead.
So I think, you might find it as epilepsy, but maybe some records might just say convulsions or like shaking or, so there may be, the phenotype may need to be broader or narrower depending on whether you want it to be specific or sensitive.
Absolutely. The next step after in OHDSI network studies, the first thing is to figure out what's the research question. And then the very next, very important step is to start now understand the phenotypes. And here is where you define your granularity of the target population. What is it? Is it anybody with epilepsy? Is it anybody with similar manifestations and so forth? So yeah. Okay, Rima had, go ahead.
So for that example, if the outcome is based on the drug that the children are given, then in the phenotype you can't use medication as like a proxy for having epilepsy.
That's a very good question. So again, it's a phenotyping question.
Yeah.
But yeah, so somebody might say, well, to identify pediatric patients with epilepsy, I might want to take anybody with a diagnosis of certain conditions, but also I might want to take patients with a treatment, any treatment of epilepsy, because that's an indication that they have the condition of interest. Now, it might be tricky, but if you limit that entry criteria of only certain drugs, then you're going to bias your results of that treatment pathway to people who already are by design required to have that drug. But if you happen to list all the different drugs, then maybe not. But it is by design. If your entry criteria is a drug of interest, then you're biasing, you're more likely to see that drug in their treatment pathway simply because they were part of the entry criteria. Any other question? You'll see when you get the treatment pathway. But one thing interesting in the treatment pathway is is the target population anybody with epilepsy, or is it treated epileptic patients? And then among those, I want to see how the treatments lined up. And usually you get the answer both ways in the standardized tools. Okay. Rima has a very interesting question in oncology.
I'm an aspiring epi, and I'm not a clinician. The question is about waiting time to the first cancer surgery. It came up during COVID because elective surgeries were delayed and the outcomes of cancer worsened, and also there was a disparity between different populations waiting for cancer surgery. And when we started researching searching for guidelines or anything definitive about waiting time for cancer surgery, we didn't find anything in the literature. So the question is the following. In the most prevalent cancers like breast, colorectal, lung, of early stages, where cancer is prescribed, a curative surgery is prescribed as the first line of treatment, so no prior treatments. What's the waiting time for this surgery? And the challenge, the major challenge for me, and I'm still trying to wrap my brain around it, is that the population is people diagnosed with cancer who had surgery, and surgery is the outcome. So Azza promised to solve this challenge once and for all.
I'll try. So we always want to describe our question in a way that we can standardize it as an input. So we want to understand what is the target population, we want to understand what's the outcome, and we want to understand the dimension of time. When you want to look at an outcome in what time frame, and we can call this time at risk. So in the context that Rima talked about, she's really interested to understand surgery as an outcome among patients newly diagnosed with these cancers. But she also wants to understand that time until these people get that surgery. So you can think about it as an incidence rate analysis, meaning that you're looking at, first of all, you have no idea. One of the questions I ask her, do you already know what's the proportion of patients who end up doing that surgery as a first treatment? And she said no. And she said that would be an interest. So first thing you want to know, among the people who are newly diagnosed with cancer, what's the proportion of patients who end up undergoing that treatment, that surgery, before they get any prior treatment? So you can think of patients newly diagnosed with all these three cancers, each of them as a target population. Do you all agree? Does this make sense? Okay, now I'm thinking in terms of an outcome. I can think of the surgery as an outcome. And I can say, among patients with newly diagnosed with these cancers, how many patients undergo treatment prior surgery? And I can think of treatment prior surgery itself as an event, as the event of the outcome. And I can define it as a surgery without blah, 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 all these other treatments. That itself becomes the outcome. And then the target is the patients with newly diagnosed. And I can define them also through a phenotype where I say you have to whatever definition of the oncology definition for the different cancers. So now I have a target population and I have an outcome population. And if I do the characterization, I will know among those who had their cancer diagnosis, and that would be my index date, I'm gonna follow them in the data, and I'm gonna find the patients who end up with doing surgery before they take any drug, because without prior drug will be part of the definition of the outcome. Does that make sense? And now I'm gonna characterize that. She's also interested with the time dimension. If you heard her, she wants to know, when does that surgery happen to occur? So now I can ask a question of, when does surgery occur in relationship to that index date of cancer? And that's a question that can be fully standardized, and you'll see in OHDSI tools, it's also one of the results that come out, and we'll look at it. It's called time to event. So you can look at where is the occurrence of that prior treatment surgery in relation to the index date of being diagnosed with cancer. She also told me that she's interested in knowing who are the patients, what characteristics do patients who end up with having the surgery different than people who didn't have the surgery or did something else, so they don't fit into that outcome. So we can also have the question that says, how are patients who undergo surgery different from those that did not? So now I want to take that patients who met the event, who met the outcome, and I want to characterize them. I want to see all those variables and see how different they are from those who didn't match that outcome, either had the prior treatment or never had the surgery. And that's something that our standardized tools also allow you to observe. Would that answer your clinical question? Perfect.
Or can you break it down to like they had surgery within 3 months of their diagnosis versus after 6 months or something really clear?
Absolutely, absolutely. You can do that as part of your outcome definition. If you have no priori, you can observe that and then start thinking of like, oh, it looks like surgery happened certain time. Let's define that and then see its distribution, 100%. As related to that, there's the idea of time. And in any observational analysis, time is very important because you have longitudinal data. So you're looking at things at different times in relation to some event. I heard people questioning, what do we mean in that handout with what's the time at risk? So when Rima said she wants to know what's the proportion of people who end up with surgery, one of the things that she needs to specify in her question is when. Does she want to look at the event, at the outcome of surgery that can happen anytime? So all the time post the diagnosis? Or is she interested in looking at surgery and identifying patients who go into surgery, particularly after 1 year of their diagnosis? So that time at which you're going to retrospectively look at the data to identify the patients with the outcome, you can limit that time to a certain time into 1 year after diagnosis, while they are actively having the disease. Or you can say, I want to use all the time I have the data. So if I have somebody in the data that got cancer and still continue having an observation time in the data set for 10 years, I want to look at any time. So that is the idea of time at risk. In your analysis, you have to specify when you're interested in an outcome, when you want to bound that outcome in terms of time of when it can happen. Many times in questions like this, if you have no idea of when the outcome can happen, you can say all time, and we call this all time post-index. If you're doing something related to an exposure to drug, you may be only interested what's the outcome while the patient is actively taking the drug. And we call that in pharmacovigilance studies on treatment, because you're looking at an outcome within while the patient is taking the drug. Or you can say fixed time. I just want to, after that index, I want to look 30 days, 1 month, 1 year, and so forth. So the time on treatment, the at-risk time is just a specification of if you're interested in an outcome among a target population, when do you want that outcome to occur? Or how long are you studying it for?
So in oncology, you also tend to have a lot of complicating factors, like you get chemotherapy before surgery, after surgery, stuff like that, right? So if you're trying, and lipotherapy in and of itself can take a whole lot of complexity. So how would you define, if it's not a fixed time, meaning 12 months, but if you're trying to say, if you're trying to answer that same question, but surgery was the first treatment they received, how would you define the at-risk window to account for all of these other
Yeah.
adjuvant, peri-adjuvant treatments?
Yeah, so that's a very good question. So sometimes it all goes down to your definition of outcome. So if you're defining surgery, that's something that happened, as long as it happened before any treatment, you can define the outcome as surgery without any treatment, like with as an exclusion criteria on the outcome itself. If you are thinking of if you're thinking of a particular time, if you're thinking that I'm going to count those surgeries, but if a person take a certain line of therapy, then they're not anymore at risk of having that surgery, you can also define that in your specification by saying basically the patient exit being in my target population if they start that line of therapy. That would be an exit criteria in your target population. You can always think of everything as a target, outcome, time at risk. And all the complexities can be a characteristics of one of those three that you can operationalize during your definition. I know we ran out of time, but we're going to go over an example in the demo that will quite much.
If you could help.
Yeah.
You could help Aniek with logging into your machine. While Aniek's getting up here, we're going to have one more presentation, and then we will take a break. But as I promised you, the slides are all available, and actually they're now posted. So If you go to odyssey.org/Odyssey2025, you'll now see that all the slide decks that we're presenting are available in case you wanted to go back and look at anything that that Marty or I said or that Aniek and Cindy will say.
Yeah, I think we're all good to go. Hi, everyone. I hope you're still awake after these 2 hours. I'm Aniek, and I'm an assistant professor working at the Department of Medical Informatics in Netherlands, Rotterdam. And although most of my work can be best described in OHDSI terms as falling under the hood of methods research or patient-level prediction, what I will talk to you about today is treatment patterns, which I can only echo actually what Patrick said earlier. I think is a really, really powerful tool to visualize and understand our data, which is important as a characterization study by itself, but also I find in my research when I'm doing more advanced analytics, such as prediction analysis, it's really important to understand the data you're working with before doing more answering more advanced questions. And I'm going to go through all these slides anyhow, but I kind of want to understand where you're all at. And usually I ask for a show of hands, but given we've already been sitting for a long time in this room, I want to ask everyone to stand up at some point in time. So the first question is, does everyone has seen this plot before? And now I don't mean earlier in Patrick's in Patrick's talk in this session, or maybe when you were in the introduction to OHDSI tutorial this morning. But please, everyone who has seen this nice plot before, can you please stand up? Okay, so that's great to see. I think there's quite some people that are familiar. I'm very happy to see this. You can sit down again. To see this number increase already over the years. You can, of course, see this one coming. So who has also really worked with these plots before? So maybe use them as a form of evidence to inform questions that they had about their data. Okay, still quite some people, thank you. And then the last question, who has ever performed a study themselves, so created evidence or these plots before? Okay, I think that's about the same group of people. So I hope for all of you, no matter where in these questions you stood up just now, I hope all of you can learn something from this presentation. And maybe next time you don't get to stand up only at this first part, but also, perform a study yourself. So what I'm going to go through in this next half hour is describe what a treatment pathway study is. So some definitions, but also why is it relevant, what can we learn from it, and what can we not learn from it. How do we then construct these treatment pathways from quite messy medical data? So understand the choices you have to make there. I'll go into how to conduct your own treatment pathway study, highlighting some of the tools that are available in OHDSI. And then on the way, I will describe some studies and developments in the OHDSI community, but this will be a smaller part given the time. But to start with, what is a treatment pathway study? So a treatment pathway itself is a sequence of treatments over time. So for example, for a certain person A, we can observe that this person has repeatedly received the same drug in equally distant periods over time. However, usually a patient record would probably look more like this. So a patient has received several drugs, different drugs with different time intervals. And here you can already see questions come up. Are we interested to describe all these 5 treatments? Do we only want to look at the changes between the treatments? Or are we only interested maybe in the first occurrence of certain treatments? So the overall goal of a treatment pathway study is best defined as we aim to find pathways consisting of treatments of interest that are defined by event cohorts within a certain study population of interest, which would be the target cohort. So you'll hear me use the terms target cohort and event cohort repeatedly. There's a lot of flexibility to define those, but I'll not go into defining the cohorts themselves. So this is the idea of a treatment pathway study. But of course, what I'm showing here is still quite an idealized version, I would say, of what you would see in an actual observational database. usually there is no such thing as 3 clearly defined different treatments. We have different formulations, we have different dosages, we have all kinds of variations, different coding. Also, these are usually not sequential necessarily, but there might be overlap. There could be long periods in between. So there's a lot of choices to make as a researcher there. And I'm going to argue that all of that can be solved by defining the cohorts that you're interested in. So really trying to define what are the treatments that you're interested in. So that's a big part of the key of doing a good treatment patterns analysis. But also being very transparent about the choices you make to then go from this medical file to your treatment pathway. So I like to start with this visualization because this is the typical knowledge gap that we see, which is there's usually a lot of research that looks at how we should treat patients. So for example, which treatment is most effective for this group of patients? There's randomized controlled trials and all forms of evidence generated to answer this question. And this question is then used by clinicians through clinical practice guidelines. So all these recommendations and the scientific work that we do, it is translated into clinical practice guidelines. So far, so good, right? But then we perform studies in our observational data, and actually the question that we don't and never answer is how are these patients actually treated in the real world? So we have the guidelines, we know what should happen, we know that these guidelines don't always apply to the group of patients that is being treated, but we don't know what is happening actually in practice. And given all the large size of OHDSI and all the observational data that we have available standardized, there's a big opportunity to start answering this question, and this is what a treatment pathway study can do. So for example, a question that I was interested in is what is the sequence of treatments received by newly diagnosed patients, adult patients with asthma across different countries? And in order to answer this question, there's actually 3 steps to do a treatment pathway study, which is first to identify the cohorts, so that both the target and event cohorts, and generate these cohorts. Then, from that medical file with identified relevant patients and treatments, we can construct the pathways. And here we need to make quite some decisions as a researcher, how we're going to go from the messy file to a simple visualization. And then all these individual pathways that are constructed from their individual medical files, they're aggregated into some kind of output. My presentation now will focus on the second and the third pillar here. So how do we construct the pathways, and how do we not aggregate the output, but mostly how do we interpret the output? Because I think that is the most relevant question for here. But to follow the example, the target cohort in this case is people diagnosed with asthma. And I have just a very basic cohort where I say I'm interested in adult patients, so the age over 18. I want to have a first diagnosis of asthma, and I exclude patients that also have COPD. Of course, these are decisions I already make as a researcher, and I just heard the question, okay, maybe we're interested in a cohort of patients that is actually treated. Treatment pathway studies do allow for that, but then you just need to be more stringent already in your target cohort. So any study population can be selected here, both more broad, both more narrow. This is what we'll use in this presentation. Event cohorts that I could be interested in for this specific group of patients are just all the drug classes that patients receive for asthma, which is, for example, SABA, ICS, systemic corticosteroids, LABA, and many more. So I'm not going to define those cohorts now, but any of the OHDSI tools that are available, both Atlas or raw SQL queries or the Hades packages can be used to both define the inclusion-exclusion criteria as well as generate these cohorts. I'm going to start from the point where we do have those cohorts generated, and therefore we can see that for one individual, their medical file looks like this. Here the rows are the different event cohorts, and this is just a snapshot of a person's history with the first asthma diagnosis in January 2016. to then move from this medical file to a treatment pathway, there are certain choices that we need to make. In the context of asthma, I made the decision to then combine drug areas of the same class within 30 days, given that here we would not expect that someone is stopping treatment and starting again, but instead is probably receiving continuous treatment where simply the medication was picked up at a different point in time. However, when there was only a short overlap in these in the drug prescriptions, these were considered a switch. So here we say the person probably changed from SABA to ICS, but still has some prescription left of the first type of medication. On the other hand, here at the end of the medical file, you can say that probably these periods where there was a longer time where both treatments were received, can be seen as a combination treatment. So now you can already see these are the kind of decisions that you need to make, and you need to have a way to both identify the questions that you need to answer with your clinical team, but also a way to communicate that you made those choices in your study design. So before telling you more about how to do that and how to use the treatment patterns package to do that, I want to show you the goal that we're working towards. So this is the plot that I showed you earlier, and I'm happy, I think many of you already know how to interpret this, which is good. Given in 2 slides, I'll have a little bit of a test for you to see what you can actually infer from a sunburst plot, which is probably like the fastest test you'll ever get after learning something new. But so a sunburst plot, it shows the treatment in the center and subsequent treatments in the outer layer. So the inner circle shows the first treatment, and then the second and third treatment, and so on. This entire plot captures the study population that we defined as a target cohort. So that is the group of asthma patients. Every color in this graph is one of the event cohorts that I defined. So the event cohorts are listed here on the side, and every color here in the graph is a different event. Combination treatments then appear as events within one of the rings in the donut with 2 events at the same time. So what can we see now if we go to the next one? Individual treatment pathways can be read from the inside to the outside, being directly the treatment received after diagnosis till the last treatment that was considered for the analysis. In the inner circle, you can, for example, see what is the first most common treatment that is used, which was, in this case, SABA, and then the second most popular treatment that was used was ICS. Well, the outer layers show the subsequent treatments, but you can already see that there's quite some fragmentation there, showing quite a lot of variability in this specific study population. And then finally, you can also see the group of patients that did not receive any treatment during the follow-up time, which is indicated by this proportion. So this is a rough summary of what you can infer from this sunburst plot. I'll just quickly show you the Sankey diagram as well, because I think it's important to realize that although this can be used to visualize treatment pathways as well, you have to read and interpret it very differently, given every stage in a sunburst plot is conditional on the previous. So where in a sunburst plot, what I just showed you, you can see the entire treatment pathway. For a Sankey diagram, you can only see switches in treatments. So between the first and the second, pause, between the second and the third. So a Sankey diagram shows the treatment conditional on the previous treatment, and you cannot go through the diagram to understand this person is then matching this person or so. So it can be useful for some purposes. I prefer to look at the sunburst plot. Then, just a very quick check to see if everyone knows what we can learn from a sunburst plot or not. I'm going to go through some statements, and if you know the answer, please raise your hand, and then we're good to go. So, can we learn the percentage of patients with and without treatment from a sunburst plot? Who knows? Cynthia, you raised your hand. Yes? Yes. Indeed, we can do that. The gap that is shown here is the percentage we're interested in. Next one. What is the most common first treatment? Can we learn this? Yes. Yes, indeed, we can learn that as well. I already gave it away just now. What is the most common fourth treatment? Can we learn this? I see some nodding here, but in the back? I guess. That is, yes. Oh, yes, okay. I was like, that sounds a bit uncertain. Yeah, so there is two things. I'm going to say no. We can, but it's difficult. Specifically for this graph, no, because there is no fourth treatment. So that is definitely the first for why it's a no. So here we cannot learn what is the fourth treatment because they're not included. But also, I want to argue this kind of visualization is much more useful to visualize the first or maybe the second, and less so if you're interested in the fourth, given you'll have to add up all these like minor fragmented parts scattered around the outer side of your sunburst plot. So maybe in some sunburst plots you can learn it simply because it's homogeneous enough. But if you're really interested to look at that, I think it's better to go look at something else. But this is a tricky one. Then the duration. Sorry?
I guess so was the right answer.
Yeah, exactly. Then the duration of a treatment. Is this something we can infer? False, yes. Thank you. This is the wrongest mistake you can probably make in this context. So I'm very happy with a good answer here. It's all about sequence and order and not about durations. Patterns of switching between treatments. Someone in the back. Can we learn this from the sunburst plot? Do you know? Can we learn this? Patterns of switching. So can we learn the order or the sequence of the treatments? Yes, we can, indeed. This is exactly what we want to do in the sunburst plot. Which treatment a particular patient received? I see nodding, which is good. No, we cannot learn this. It's anonymized, and we cannot relate it back in any way to the original data. And the amount of time between treatments. Is this something we can infer? No. Then the final one, which treatments were given simultaneously as a combination? Yes, this is something we can learn. So, given you all passed this test, I'm already very happy because it's very important to be able to read these plots properly if we're doing these analyses. So, just to link it back to what we can actually learn from it at a slightly higher level is using these treatment pathways, defining that, reading the sunburst plots, we can actually infer what is happening in current treatment practices. So, and we can use this kind of information to, for example, better design randomized controlled trials, because only if we know which treatments are being used, we can actually define good comparators in randomized controlled trials. Also, it gives a lot of opportunity, for example, to give insight into treatment and doctors' preferences, both within facilities, but also across facilities with similar care settings. And finally, which I already argued in the initial figure I showed, is that it's very helpful to improve the implementation and revisiting of clinical guidelines because it helps you understand if the guidelines are actually followed in practice, or if we're so if we have to target that part of the cycle, or if we have to do more work and figure it out what we need to do. So using this large-scale characterization can help to give these high-level insights, and also when showing these results, it often gives a lot of hypotheses for follow-up studies. So for the clinical question that I just showed you, the example I used, I actually performed a network study using OMOP databases, and these were the results. I'll not go into this now, but for those interested to explore, take a look at the shiny here or the publication underneath. I think it's very interesting to look for me at these kind of plots because immediately already from a distance you can see that they vary a lot. and then you're able to have a conversation on why is that and what differs. So I just want to highlight that within OHDSI over the years, quite some publications have been done for treatment pathway studies, and I hope that many more will follow in the coming years. So please take a look if you're interested in any of these domains for the results, and think about possible studies that you could add to this. In order to do so, I'll finish by going through the tools that are available within OHDSI to do these kind of analysis. Which is on the one hand Atlas and the Cohort pathway functionality, which some of you might be familiar with. This is most beneficial to do very quick and standard analysis on treatment pathways. It is included in the Atlas interface, and just with a few clicks you can actually already get this sunburst plot, which is, I think, powerful for a first exploration if you're interested. However, if you want to do a treatment pathway study, I would definitely recommend you to go to the treatment patterns package, simply because it's much more elaborate. There's much more freedom as a researcher to choose how to form the pathways, and there's much more output also that you can use for your study. And this treatment patterns package actually resulted from the very first study that there was on treatment patterns, which was very impactful in terms of how big the scale was of this first analysis. It immediately looked at hypertension, type 2 diabetes, and depression, if I'm not wrong, right? So very impactful, and actually triggered the question of, can we not do this for other disease domains? But then also immediately the question comes up, how do we make such analysis scalable? And this is why within the Eden project, I started developing the treatment patterns package to actually be able to scale up this kind of study and also to make it reproducible. Because trying to replicate a study with so many choices, it's really important to be clear on what happens. So that was the beginning of the treatment patterns package. And right now it is maintained by Maarten, which is a colleague of mine. And thanks to him, the package is now in a very great shape and can be really used at a large scale. So it's been co-developed within DARWIN-EU and has made it by now to both the HEDIS suite as well as CRAN. So for any of the package documentation, I would definitely advise you to go to visit one of these links. But for now, I will discuss a little bit more about the package internally and what it tries to do. So it tries to formalize the entire process of going from the medical file to a treatment pathway. And so in the original publication, what we did is identify all the choices that you need to make as a researcher. I'm not going to go through all of those, but I will show you, I think, 3 or 4 examples of choices that need to be made and how you can opt for the right value. It has been further developed within DARWIN-EU, and so some functionalities have been added, and some you'll see maybe some parameter names have been changed. But it's very interesting to look at the GitHub and see all the possibilities there. But I'm going to go right back to the beginning, which is if we want to do a treatment pathway analysis, we're actually interested to analyze events during a certain target period. This target period is defined based on the target cohort. So your target cohort study population always has a start date and some time, continuous observation time, so also an end date. We're interested to look at all the events that occur within that period. So this is the first thing that's important to realize. So these kind of events would be included. However, here the event at the start in green would not be considered in the package. That is for the basic settings. There are settings to then vary this and to maybe include this event as well. But in principle, we're aiming to make a pathway of events that occurs in the target, within the target time frame. Choices that you then need to make is, for example, the min era duration, which is how long should an event take, so how long should the treatment persist in order to be included in the pathway. So a value of 0 would mean all the events are included. A value of 30 would mean only events that take longer than 30 days are included. Now you might say, why do we remove events? Well, if there's many short periods of treatments, you might not actually learn that much from your treatment pathway analysis. So it's all about finding that balance between the nitty-gritty detail and like being able to visualize and exploit like the main, most important trends in your study population. So just a short example here for this person 1, with a minimum duration of events of 5 days, the treatment pathway would be AB, because both events are longer. For person 2, we would skip the first event, the A, because it's shorter than 5 days. For person 3, event B would not be included because it's too short, so we would focus on the main treatments that are received, which is AC in this case. And then something that is important to realize, with these settings, it would mean that person 4 would end up as being not treated. So if we would have a 0 value here, then we would say this person received treatment D, but now we say this person is not treated. I'm not here to claim what is right or wrong. This is the impact of the choice that you make as a researcher, and I would argue this is one of the reasons why you want to do sensitivity analysis. If I perform treatment pathway studies, I usually perform sensitivity analysis with different types of, like a very broad, very lenient, and very stringent choices for these parameters, which would be able to give you some kind of range of this non-treated population to see if this is actually, you know, influencing your estimates if you're looking at treated versus non-treated. Then there's another parameter which is called error collapse size, which allows you to merge event eras that are close to each other but similar. So here you specify the duration between 2 events of the same cohort and when they are considered as one and when not. So I'll just go to the example immediately. Here, looking at this person 1, it allows you to focus on AB instead of A1, A2, B. For person 2, this would mean it translates to ABA. And for person C, because you chose a number of 5, you still see that there is a repeated treatment. Because here the period between is longer, and so we actually really infer this as a stop in treatment and a start again. Of course, this is dependent on the type of disease you're studying and if this is desired or not. And then the final parameter I want to highlight is the combination window, which is the duration of how long 2 events need to overlap to be combined. If the overlap should be infinite, then there will be no combinations. And if the overlap is 5, so if the combination window is 5, then any 2 events that already have an overlap of 5 days would be combined. Well, to see how this plays out, for person 1, the events are not overlapping, so of course there's no combination. For person 2, there is substantial overlap and they would be formed as a combination, but you can also see that the period before and after is still considered as a phase of monotherapy. There is another setting in this package, which is called post-combination duration, which allows you to specify how long these events should last in order to include them or not. You can already predict it, yeah. And then the final one here, the whole phase would be AB. So just to make my point here, dependent on the type of disease you're studying, these settings would differ. So for example, for a condition like hypertension, which is more long-lasting, and you would expect a longer follow-up, it makes sense to have slightly higher values for these parameters. On the other hand, for a very acute disease, like in any type of infection, it makes more sense to look at a shorter period. So this is how your knowledge that you have of the clinical domain should impact all these study choices. Just to highlight some other possibilities with the current package is anchoring, which is what I already referred to a little bit earlier. For the package, the starting point is we're looking at the periods of the target cohort, so the start till the end date. But it might be that for some cases, and actually it was the case for our asthma example, sometimes treatment is initiated to confirm diagnosis. So for example, for asthma, we know it's very common that people are prescribed some asthma medication to actually see if it benefits them. And for these kind of cases, you might not want to start at the moment of the diagnosis, but you actually might want to start just before. So there's this thing called anchoring, which allows you to both before and after vary the time periods. Then we have something for acute and therapy split. So this is more the long-term and the short-term duration of events. And also it is important to realize that some people might leave your cohorts due to other external events. And so for diseases where it might be more prevalent that someone dies, unfortunately, it might be important to have exit cohorts so that you can also see in your pathway analysis when someone exits the cohort or when someone simply doesn't receive treatment any longer. Because remember, we're only visualizing the changes between the treatments. And so if someone is only included for a shorter period in your study population, you might incorrectly infer that as someone only received one treatment, whereas actually someone exited the cohort. Just to quickly highlight, I want to draw your attention to the fact that Atlas and the R package have a different way of using a combination window. So when we're interested in studying treatments that occur simultaneously, there is a difference with how the two handle this. I don't necessarily need to go into the details now, but the Atlas, it's more related to the moment of the start and end dates. And the combination window is related to the duration of overlap. This is, with any choice, there's not a general good or bad answer to this, but these lead to different results, and I just wanted to bring that up here so you're aware if you're moving from the Atlas package to the R package that there is a reason for why your analysis might differ. Then, some best practices from doing these treatment pathway studies that I want to give you along if you want to do your own study. I've organized them in the way of developing cohorts, construct pathways, and aggregate outputs. Is on the starting point of this type of study is that there should not be too many or too small cohorts. Of course, if you're going to define, let's say, 50 event cohorts you're interested in, your pathways are never going to be comprehensible. So I think a good amount is usually to aim for 5 to 15 event cohorts, because then you have something that you can probably comprehend as a human. Also, usually including too small cohorts is not very relevant, because you're going to look at a very specific kind of niche subject, where I think the main power of treatment patterns is to study the overall trends, patterns in your study population, both in subgroups as well as over time. Also, when defining the cohorts, it's very important to think about a minimum observation time for your target cohort, given that we're really studying the pattern of events within that duration. It is important to make sure that someone is long enough in your database so that you actually have time to consider. Then for the construct pathways, there are the 3 choices that I just highlighted, but there are more choices. There's more freedom, which I think is a good thing. But there it is important to go through those choices with your clinical team and consider the nature of the disease, for example, as I showed, but also, for example, the order of diagnosis and treatment. And then finally, for the aggregate output, focus on the main pattern. So don't look at the pathways with a low outcome count. They should usually not be considered, and there's also a setting to minimize those. Then it's very important to do some kind of robustness analysis by varying the study settings, as I explained earlier. And I think the trick also when looking at these results is to understand you need to do follow-up studies to answer the questions that will come up. Not all questions will be answered by this visualization, but my experience is a lot of questions are triggered by the visualization. So write those down and go do the studies. That is, I think, the power of, of this kind of analysis. So some quick takeaways. A treatment pathway is simply a sequence of treatments over time, no duration involved. It can help improve clinical practice in various ways by informing clinical trials, understanding how doctors are prescribing, etc., how good clinical guidelines are followed. And if you want to do an analysis like this, be transparent about the design choices you make. And therefore, using the treatment patterns package is a very powerful way to do that, because it allows you to communicate the values you chose for all these different study settings and compare them across. So, next steps and how can you contribute? I think we're in a very powerful position to do treatment patterns analysis at scale. And so it would be great if in maybe a year or in some years, we really have treatment patterns for all types of patient populations across many drugs, across many countries, to better understand how clinical practice is performed. And there's also many interesting follow-up research questions related here to the duration of treatment, how to automatically select the relevant cohorts, and many more. So if you're interested in this, do come talk to me or Maarten, for example, and very happy to chat. Thanks. I think now, housekeeping-wise, we have a break. Patrick,
So
can you? Yeah.
I'm going to say that we'll be back at 3:30, so we'll give 10 minutes to encourage everybody to go stretch their legs. I think there may be like a drink out there or something, and come on back at 3:30, and then we'll continue with cohort incidents.
Great.
Hello, everyone. My name is Cindy. I'm a PhD student working with George. And this is going to be about cohort incidents because somehow within the last, 3 years, I've accidentally branded myself as the how often girl, which I'll talk a little bit more about as well. Okay, so here are some of the goals I set out for this session. So by the end of the session, you should know what incidence rates are, learn a little bit about what OHDSI has done so far with incidence rates, and lastly, a little bit about how to potentially implement your own incidence calculations, though I only go very briefly about this topic. So if you're actually interested in implementing some of your own incidence rates calculation and you don't know where to start, hit me up or hit Patrick up. Okay, so incidence rates calculations are one of the most common analyses in pharmacoepidemiology. Whether it's used for comparative background rate estimation, for drug adverse events, or assessing the potential impact of adverse events for designing clinical trials. And this is the definition of incidence rates. It's defined as the ratio of number of new outcomes within the defined time at risk over the person time within the population at risk. And here, it's important to note that within the OHDSI kind of tool stack, we can calculate incidence rates and incidence proportions really easily, but the two are not equivalent. And it's an important concept that they are different. So incidence by itself, or the incidence proportion by itself, it's not enough to tell you completely what's going on. It's not the full picture. And here, I just want to point out that the numerator and denominators between the rate and the proportion is different. Both the numerator and the denominator is different. So the rate is new occurrences over time at risk. And then the proportion is kind of like a percentage, like people who have the event over the people at risk. And people like using that because it's a percentage, it's easy to interpret. But what it doesn't account for is time. And rates normalize over the amount of time that you have observed in your population. Okay, and this is just my note that both are supported within the OHDSI tools in the cohort incidence package. Okay. So why incidence rates? Because it is so simple. But it is because it's simple that we can use them. Basically, it has fewer assumptions. So people are, obsessed with machine learning, causal inference, and those things are great. But a lot of times they come with a lot of assumptions that may not, that makes everything more complicated. And Patrick actually has this really cool graph of all the evidence gaps that we currently have in medical literature, which I don't have, but I would love a copy of at some point, Patrick, if you, I'm gonna talk to you later. Basically, Patrick has this really cool graph of all the possible hypotheses between like outcomes and drugs and the real evidence that we have. We only have like specks of evidence. This graph is basically blank because we have so many gaps. And incidence rate have the potential to fill these large gaps in evidence. And even though it's not causal, it can still be useful. So this is a simple example. So let's say we're interested in like a drug and a side effect. Now, if the incidence of that side effect and that drug is low, then we're good. We probably don't need to worry about it anyways. Now, if the incidence is high, we probably still need to look out for this outcome, even if it's not caused by the drug. So regardless of whether it is causal or not, it's still providing us with some helpful information. Over the last couple years, OHDSI has done a lot with incidence rates. So this has been mentioned before in Patrick's intro slides. OHDSI has used large-scale incidence rate studies to provide information to both the EMA and the FDA related to adverse events of interest from the COVID vaccine, so helpful for medical guidelines and things like that. And methodologically, this is a paper by Anna Ostropolitz. OHDSI has also looked at different methodological issues as it relates to how we compute incidence rates. So, for example, in this paper, Anna found that incidence rates and their calculations are sensitive to things like start date, anchoring, age, and database, and these stratifications are important for how we interpret our results. And in addition to these methodological contributions, OHDSI has also demonstrated that we can calculate, incidence rate at some kind of scale. And that's why one of the things we decided to take on at the 2023 OHDSI Symposium is what is called the How Often study. And what we did with the How Often study is, if you were here in 2023, pre-symposium, like over the summer, we did tons of community calls, rapid protocol drafting, phenotype development, and really engage the whole community to come up with research questions and help design the final analysis package, which was then run in the symposium over the weekend in 2023. So it was really cool getting the community all come together, contribute, come up with research questions, and eventually getting together in person at the symposium to actually run the study together. And it actually wasn't only one study, it was actually 6 different studies because there were 6 different research questions, 6 different questions, 6 different studies, 20 different research questions, which resulted in us generating over 200 different phenotypes. These studies were run on 16 databases over the course of that weekend, and in all we generated 54 million incidence rates, which are also stratified by age, gender, index year, and database. And of course, because this is OHDSI, we always have our results publicly available. And if you go to results.ohdsi.org, all of the studies that are titled How Often Blank are results from the 2023 Symposium. And so from there, I want to highlight one particular study that we did in the symposium, which relates to how different drug indications affect incidence rates. So this is just a little taste of what came out from the symposium. So as you recall, that one methodological study by Anna showed that stratifying by age, sex, and database might be important for our incidence rate calculations. But the question that we posed in the symposium was, should we also actually stratify by drug indication? Because some drugs, have multiple indications, and when we calculate the incidence rates of different outcomes, is it also important to say, okay, this was a beta blocker used for hypertension versus this is a beta blocker that was used for after a heart attack. And so to do that, we had 8 different drug classes with more than 2 indications. We looked at 73 different health outcomes, and over the course of the symposium, we calculated 75 different estimates across 13 different databases. And what we found, this is just a snapshot of the results. For space purposes, I did not put all 8 different drugs and their indications on here. So the top row are beta blockers and their different indications, which are hypertension, heart failure, and myocardial infarction. And then on the bottom, there's 3 different drugs with 2 different indications, so trimethoprim, SGLT2, and GLP-1s. Okay, so let me just explain what's going on here because it's kind of busy. Each histogram is incident rate ratios So incident rate ratios will compare the incidence rates between two different indications. So for beta blockers, for example, in this graph A, you'll see hypertension and heart failure. And so how to interpret this is that if the incident rate ratio is close to 1, that means the incidence rate for that particular outcome within those two indications are pretty similar. Meaning the incident rate for, I don't know, vomiting, let's say, is similar between beta blockers used for hypertension and beta blockers used for heart failure. And so if you look at this graph F here, GLP-1s for type 2 diabetes versus obesity, all the incident rate ratios are kind of clustered around 1, meaning that for GLP-1s, the indication doesn't really matter that much when it comes to the incident rates. Whereas if you look at something like this, beta blocker for heart failure versus myocardial infarction, you see this, for example. Oh, you can't see my cursor. I've been doing this the whole time. Okay, now, you see this right here, which shows that whatever this particular outcome is, the incident rate is pretty different between beta blockers used for heart failure versus beta blockers used for myocardial infarction. Okay, all that is to say, we looked at these incident ratios for different factors like sex, age, database source, and also indications. And I've kind of just summarized the different incident rate ratios distributions here. And because it's kind of busy, I just want to draw your attention to the very last column. So this is the percentage of incident rate ratios between 0.5 and 2. And basically, I calculated this as a relative measure of how often a specific factor has less than a doubling effect on the incidence rate. And so what we can take away from this column here is that age and database, so these 43%, more often had a doubling effect on the incidence rate. So they affect the incident rate more compared to indications and then sex. So the kind of the relative importance of these different factors as it affects incidence rates is age and database are often most important. We should account for that. We should stratify by it. And then indications, and then sex. Okay, so this is just a really quick overview of this project and what has come out of the symposium and the study that we ran there. And hopefully we'll be submitting the paper very soon. Okay. So what else is on the docket? There's something called the How Often All by All initiative. This first started a while ago, but I kind of picked up a little bit of the work once I joined Columbia. And the idea is that, we did the OHDSI Global Symposium. It's proof of concept for very large scale and rapid incidence rate generation. But how often all by all, we're attempting to generate incidence rates for every drug. So I think there's 2,600 of them based on RX norm terms, every drug and every side effect. So every side effect, we're thinking about 18,000 different side effects, which would generate about 40 million pairs of incidence rates. And we would have to also additionally stratify by age, sex, because they're important, and potentially index here as well. And why are we even doing this? Because this is such a big undertaking, like generating this many incidence rates. Even though the calculation is simple, generating at scale does prove to be a bit of a challenge. So why even do that? Well, I said earlier that we have these huge gaps in evidence. And what currently exists is we have prospective studies and randomized control trial, of course. That's kind of the gold standard when it comes to generating medical evidence. And we like that because it's potentially causal, but they're really hard to do at scale. We're not running 40 million RCTs to answer our questions. It's hard to do that. The sample sizes are also generally smaller. Right? They can also be less generalizable because of strict inclusion and exclusion criteria. On the other hand, we also have drug package inserts, sometimes based on post-marketing surveillance. They're not causal, they just depend on what people have reported. In fact, because it depends on what people have reported, they may have nothing to do with the drugs. And few have rates. And there's also guessing, which is, another way to say expert opinion. So doctors and their previous experiences, what they remember of what they've read, and their own theories. And so how often all by all, these incidence rates are not meant to replace prospective studies and randomized control trials, but they're trying to fill the gaps that are currently being filled by drug package inserts and guessing. So we can do a little better than drug package inserts and guessing, is what I'm trying to say. And there was a proof of concept of this how often all by all back in 2017, and this was the landing page of howoften.org, and it was run on the Columbia database, and as you can see here, you just like type in like what drug you're interested in, what condition you're interested in, and you get some rates. And this was used back in 2017 internally within the Columbia NYP Emergency Department. It was used by some staff, but George said, honestly, it was used by one person who knew what informatics were. And the idea is that if someone came in with some bizarre symptom, you don't know what's happening, maybe you can check for the med list and check this resource and see, maybe come up with some hypothesis. But in the end, there was no real formal evaluation. And Elise Ron, who was a clinical informatics fellow, also in the HRPSAC lab 2 years ago, she did some validation of the incidence rate. And she found that it was pretty reliable, though it did really underestimate sexual dysfunction. We did not maintain this proof of concept, however, and OHDSI kind of just moved on from it in 2017 with other initiatives, but we are really hoping to bring that back. So if this is something that you might potentially be interested in getting involved with, let me know. Love to talk. Okay, so that is all I've prepared for the OHDSI's journey part. So the next important question is, how can I run my own incidence rate study? And the really simple answer is that we have a package for it. Just like most things in OHDSI, we have a package for it. And this package is the Cohort Incidence package. And if you're familiar with the Strategist toolset, this package is also within Strategist, and I find that that's the easiest way to be able to hand off your code to someone is run something through Strategist. So if you're familiar with that, great. If not, don't worry about it. And I broke up kind of running your own incidence rate study into 4 different steps, sorry. First is formulating your characterization question. Second is to generate your cohorts with OHDSI Atlas. 3, use OHDSI tools to generate your incidence rate. And I just listed a couple different considerations here that you can think about, which I'll talk about as well. And then lastly, view your results. Okay, so starting with formulating your study question, this is something you saw with the handouts. So this is just a, formulary kind of research question that you can form. So I just put an example here. Among patients who are new users of ACE inhibitors, how many patients experience angioedema within 1 year of starting the drug? So that's something you can answer with incidence rates. Next step is to generate your Atlas cohorts. Unfortunately, how exactly to do this is outside the scope of this talk. But if you've never done this before and you want to know how, I'm really happy to help. And I'm sure, there are tons of people within OHDSI that are better than me than this and may be able to help you better. But that's the great part about OHDSI, is that everyone is so nice. So here I just put a screenshot of my ACE inhibitors and angioedema cohort. So yeah, anyways, this is also outside the scope of this talk. Step 3, create strategist specifications. So like I said, Strategist is a package that allows you to design and execute studies against the OMOP CDM. It also includes other packages, so it's kind of supposed to be a workflow where you can do something from diagnostics to characterization to population level prediction, et cetera, et cetera. But what you can do actually is comment all of those other packages out and just run the cohort generator and cohort incidence, and then that will get you what you want. That's what I usually do if I just want to run an incidence study within Strategist. This is a screenshot of some of my code, just importing the cohorts that I generated through my Atlas. So these are Atlas IDs right here. And this is what it would look like, how you would integrate Atlas within your Strategist. And there's a file called downloadcohorts.r in Strategist that will kind of set this all up for you. And I honestly really recommend getting familiar with Strategist, even though this is outside of the scope of this talk, because it does make everything really, really easy. Okay, but more importantly, there are some code inside Strategist and also the cohort incidence package that are more relevant to creating your own incident study. And these are 3 things: the clean window, the time at risk, and the subgroups. So I'll start with time at risk. Time at risk is when might the outcome occur? Like, how long do you want to follow the people for to consider something happening to be an outcome happening? 3 examples I have here. You can say, okay, at cohort start, that's when they start taking the ACE inhibitor or whatever drug, and then I'm going to observe them until cohort ends. So this is called on treatment. You're going to observe someone until they stop taking the drug. Or you can say, I don't really care like whether or not they're on the drug. I'm going to observe them for 30 days after they start taking the ACE inhibitor. Or I'm going to observe them for 365 days after they take the ACE inhibitor. So that's another way you can define this time at risk. And lastly, there's intention to treat. So you can say, okay, I'm going to start observing them, and they can have the outcome occur anytime between when they start taking the drug to when we stop observing them in our database. And that's intention to treat. So that's 3 examples, and this is something you can set yourself within your code specifications. Another thing is the clean window. So the clean window will impact when we have recurrence of events. So for a chronic disease like diabetes, if you have it once, you can't ever be at risk for it again. So you might set the clean window as 9999, meaning that if they have it, they can't ever have that outcome again. Now, if the event can reoccur, the clean window is basically the period of time between 2 successive events, like how you want to distinguish it from just regular follow-up care and the first time it occurred versus the new time it occurred, and when you want to consider it an outcome. So for example, if your outcome is acute myocardial infarction, you might want to set the clean window as a year. Because after a year, whatever previous AMI they had, that's probably the old AMI, and now it's a new occurrence. Versus something like a cold, which is more common, you might set your clean window to be like, I don't know, 7 days, 30 days, something like that, because maybe within like a month, that's not the same cold, and it's a new outcome that you can consider an outcome within your incidence. Okay, so if that sounds a little confusing, because I explained it poorly, basically the clean window just ensures that when we count a patient's outcome event and we see that outcome event occurring, it's actually a new incident event and not something that is part of a previous occurrence. And lastly, for subgroup considerations, strategists and cohort incidents actually can easily stratify incidence rate by subgroups of age and gender. And actually, by default, it is just stratified by index year, age, gender. If you want something a little more complicated, like let's say you want to do the drug indication study that we did with the incidence rate, you might have to either create each subgroup as a separate cohort within Atlas, or do something a little more complicated within Strategist to make sure the subgroups actually happen. And lastly, to view the incident rate and incidence proportion results, Cohort Incidence or Strategist actually outputs something called a results folder for each database. And what you want to look for in this results folder, so I know the text is probably kind of small, but this folder, it's all standardized. So it'll go under results, your database name, strategist output, and then cohort incidence module. And the file you want to look for is CI incidence summary.csv. This is all just standardized within the package. It's just how the package was written. And then you'll see a CSV file. And what you're going to look for is these 2 columns. There's one column called incidence proportion per 100 persons, and then an incidence rate per 100 person years. So that is essentially what you're looking for. And I know this is like kind of vague, probably, but that's kind of it. And I say sort of because this is just a really quick overview. I did not prepare necessarily the depth that is probably required for you to like go from 0 to like running your own incidence rate, but hopefully it kind of gives you kind of an idea. But also I'm happy to help anyone if this is something that you're interested in implementing. And if I don't know how, I'm sure Patrick knows how. And that's all I have for you guys.
Thank you, Cindy. Any questions for Cindy? Comments? People just wanting candy? Cindy?
Yeah, 40 million sounds pretty daunting. Have you thought about just like, like a list of the top 1,000 or something, Because then a lot of people could run that and show it to people like at hospitals and like regulatory agencies and, at least whet their appetite?
Yeah, the 18,000 outcomes is actually already a pruned list of SNOMED outcomes. But actually, I think the last time I tried running this with the 40 million, it only took like 2 weeks. So it's not that bad. Yeah.
I'll just add, actually, I think we're at a really interesting inflection point in terms of what it takes to do large-scale analysis in the OHDSI community. At this point, it's not actually a problem to design a study that could do a large number of analysis or to execute an analysis that'll generate a large amount of analysis. It's actually the challenge of once we have the results, what do we do now? And I think that's actually an interesting problem to have that I really want to challenge our community to think about is for most everybody else in the world, it would be impossible to imagine creating that kind of evidence. Now, as Cindy's saying, like it's not only is it not unimaginable, it's actually like pretty reasonable to do. But the opportunity then sits there is like, now we're talking about more evidence than the world's ever seen. What are we going to do as a community to actually make sense of it?
Yeah, I think one of the considerations that we had, and this was brought up at the OHDSI Symposium as well, is if we were to release some tool, like how often to the public, like how do we actually communicate that these rates are not causal? Like we have to be really careful with this sort of like dissemination of these rates.
Nice, yes. Yeah, so I have a question more technical, maybe about this 40 million. So you have these drugs and the outcomes. Are those purely based on the codes, or do you make like separate concepts for the individual drugs and individual outcomes? Because this is a lot of work if you have to also define all the different phenotypes.
Yeah, it is just based on the codes.
Okay, yeah. Nice, thanks.
You can have a candy, Tom. All right.
How often the studies we did in Symposium 2 years ago, those were all phenotypes. The outcomes were fully phenotypes and the targets were phenotypes. So yeah, the community members submitted phenotypes to the OHDSI phenotype library and they were all full phenotypes.
Yeah, there's a difference between like how often Symposium and how often all by all. And I've been like, I created these graphics, but I was like, okay, it's too confusing and nobody cares.
Thank you, Cindy.
And there will not be a quiz during my talk now. So if you want to earn more candy, then you do need to raise questions. So feel free to ask questions during this. So on to the deep dive in risk factor analysis. Let me just immediately jump to the outline of this next half hour. I quickly want to set the ground for what are we talking about when we talk about risk factors. So some definitions there. What is a risk factor? And specifically, what is not a risk factor? So we're not talking causality here. So thanks for bringing that up already. What is the goal of a risk factor analysis? How do we see that within OHDSI? What is the relevance of looking at risk factors? But then the most important part of the talk and what I want to have enough time for is the second part. So how do we identify risk factors in real-world data? Specifically, what should we not do? Which is maybe not the best place to start, but I do want to end then with how should we use the OHDSI tools and some best practices there. So do feel free to ask questions if you have some to earn some candy or to, get your question answered. So what is a risk factor? So a risk factor is any characteristic, exposure, or condition that increases an individual's or a population's probability of developing a particular disease or negative health outcome. So I specifically want to add here, it's a variable. I define it as a variable that's associated to the outcome. It's not a causal factor. So some statements that people might make to claim risk factors is the risk of stroke increases with age. Something that someone could say. The risk of stroke. Smoking is a risk factor for lung cancer. Or you could say people with diabetes have a higher risk of cardiovascular disease. These are all targeting risk factors. But as I already said, but I am going to repeat it again, a risk factor does not imply causality. And why do I say this so often? Well, it's something that people know, but there's a difference between knowing and internalizing this. So please, I hope at the end of the talk, if you take away anything, it is not a causal factor. There's many different types of risk factors, and I think everyone probably has some examples in mind when you think about correlation or association is not causality, but this is one of my favorites. It shows a strong correlation between the number of ice creams that's sold and the number of shark attacks, which very strongly shows, of course, that this is not, there's no causal relation between those two, but there is a strong correlation. I'm sure all of you know examples of, you've probably seen examples that show this, but I want to dive into a little bit of detail about why are risk factors not causal and what are actually all the different types of relations that correlated variables can have. And you don't have to remember all of these by the end of the presentation, but like thinking about the different types of relations that there can be between variables is very useful to understand why there can then be a correlation that is not a causation. So for example, we can have a risk factor that has a direct causal effect on the outcome, so the direct causation, but it could also be a reverse causation. For example, if we think of a risk factor as being painkillers and the outcome as being a headache, then we can think that maybe using an excessive amount of painkillers might actually induce the outcome sometimes. However, of course, when you have a headache, you would also take painkillers. So between those two, taking painkillers and a headache, there will be a strong correlation. However, the relation might be causal or it might not be causal. Well, adding another element to it, so a third variable, there could be a correlation or not again. So for example, we can think about another factor that influences both using painkillers and the outcome. Say any type of chronic disease. Maybe someone is in pain and therefore is taking painkillers. But also headache might be a symptom of this chronic disease. In that case, there is a correlation. However, maybe the painkillers were not taken for the headache. Maybe they were taken for some other outcome that is part of that chronic disease. There could also be a collider. So if you study this set of variables and you're, for example, looking at people that are missing work very frequently, you might actually find a relation between the painkillers and the headache if you would condition on that variable and therefore maybe find the correlation and then still it is not a causal effect. Well, then of course this effect between the painkillers and the headache can be mediated by different types of variables, such as maybe the sensitivity to this whole, or maybe certain genetics, or maybe sleep is an intermediate variable in this way, or it could be moderated by a third variable, where it is actually even possible that you do not find a correlation between the painkillers and the headache if the variable is strong enough affecting this relationship. And finally, there could just be some kind of random spurious correlation in the data that makes sure that there is a correlation, but then still it's not a causal effect. Now, I understand these are a lot of relations, and I'm sure some of you have seen this before, but the point I just want to make is there's many different relations possible for correlated phenomena, and so a risk factor is not causal. So what is it? It is associated with an increased risk. So it's more saying, are the two related? Then does one change the other? To assess causality, say you would be interested in that and not in risk factors, then we need more advanced study designs. It is possible in observational data, but if that is what you're interested in, it is important to look at the cohort method package, and I'm not going to talk about that in this tutorial. But this is not required to do when you want to identify risk factors. So the goal of a risk factor analysis is to identify what baseline characteristics occur more frequently among those in the target cohort with versus those without an outcome. No questions yet? Okay, then I will continue. Just a quick, brief intermezzo. So there's different types of risk factors that you can think about. Of course, there's risk factors that can be modifiable, and there's ones that are unmodifiable. You might all come from your own clinical domain and expertise, and so there might be several risk factors that are known for certain outcomes, but there might also be yet undiscovered or risk factors that are less well known. And knowing about these risk factors is of interest for several reasons, but for example, for modifiable risk factors, it is because they perform targets for intervention. You can formulate prevention strategies, you can educate people on risks, make sure they change their behavior. But even when risk factors would be unmodifiable, it is arguable that it's of interest because then it can be used for early screening to maybe optimize treatment strategies and for at least timely knowledge and action. So in the overall medical literature, there is a lot of interest for risk factors. I've not done a proper study on this, but I think it would be interesting to see like how many papers have some kind of risk factor analysis included in there. This is not a very random sample that I put up on the slide here. These are all papers that are looking at risk factors, but they're specifically all using some kind of data-driven methods, data-driven algorithm or machine learning strategy to identify those risk factors. And actually, this is what I'm going to talk about now, which is why not to do that. Why is prediction not the way to go to identify risk factors, and why might it be better to do large-scale characterization instead? And I'll try to give you that message by showing some of the work I did myself, presenting you some of the OHDSI tools and how you can use them, and giving you the nuances of these different tools and how you can combine them in the end. So just to recap, the goal here would be to identify variables that increase an individual's or a population's probability of developing a disease or an outcome. And for this, observational healthcare data has a lot of potential, both to identify such associations, but also to then estimate the strength and the significance of those associations. And the goal is to identify all such variables, I would argue. Both confirm the known variables, because you do expect to see those back in your data, but also to discover new variables. Of course, it's important to say here that the variables should be available in your data source. If it's not in the data source, you're not going to discover it. So it's limited to, of course, the data that has been collected. Now, some use prediction models, such as, for example, lasso regression, to develop or to interpret for risk factor discovery. And when I say develop or interpret, some just really develop a model to identify the risk factors. Some develop a model because they're interested in prediction, but then still interpret that model in the end to identify some risk factors. So you can already, at the start of a research study or project, make the decision, or you can, after the fact, of course, interpret it in such a way, and both are problematic. Of course, I'm giving away some of the message here, but there are also reasons for why you might think this is a good idea to do. So why is it a good idea? Well, a large-scale, data-driven approach really allows to identify new risk factors. machine learning algorithms are known to handle big data very well. They can learn patterns from the data and, identify some variables that are important. So there's also good reasons to think that this is a good strategy. But what is the problem for risk factor detection? Well, in the medical domain, it is this problem. Well, procedures, medical conditions, and drugs often co-occur. And you might all know this, but I think it's important to realize that this is really impacting this specific problem. So, procedures are often used to diagnose medical conditions, and when you're diagnosed with a specific medical condition, you'll get certain treatments. So that means that there's a very high correlation between these different variables. Now, if you're going to train any prediction model and you're really interested in prediction, then you might use any of these variables to get an accurate prediction. And therefore, machine learning algorithms might ignore some of these variables. For example, if you're trying to predict the risk of an asthma exacerbation, you might use some kind of procedure indicating a more severe asthma symptoms, or you might use medication that was prescribed for severity of asthma symptoms. So there might be different variables in your data that can be used by this model to get a high prediction. So the machine learning algorithms, they just minimize the error. They just minimize the sum of squared residuals. So I'll now quickly go through this work that I did a couple of years ago to illustrate the potential issues, and we did so by investigating the stability of models developed using lasso logistic regression. And whereas stability from a machine learning perspective is often looked at in the form of resampling, so given I have my data, I do some bootstrap analysis and then see how it is affecting the final model. In this large-scale experiment, we did a similar thing, but then with phenotypes and study design settings that are actually impacting our decisions when we develop prediction models. So we did a large-scale experiment on real-world data, and we developed more than 450 prediction models. We developed approximately 50 models for 9 different prediction questions. So each of these questions was predicting an adverse event of interest for COVID-19 patients. And within each of these 9 prediction questions, we then used more narrow and more broad phenotype definitions to characterize the study population, to characterize the outcome of interest, and we run it across different databases. So this is how we get to the 50. Having developed all these models, we then measured what the variability was in the actual models. So we didn't look at the performance, or I have to rephrase, we also looked at the performance, but that is a different study in a different paper. For this work, we looked at the actual model that was used to make the predictions and see how those vary. So the first question we asked was like, how many variables were selected in each model? So counting the number of variables. Then the second was which variables were used to make the prediction. And the third was how did the sign of all these variables differ between the models? So we found substantial variation in the selected variables across these models, and I'll now show you the results in a little bit more depth. So on this figure, you can see the stability as we measured it by an estimator that's common in the literature, which has a scale between 0 and 1, where 1 would be stable and 0 would be completely unstable, and the x-axis here is the number of outcome cases. So the first thing you see, of course, is as the number of outcomes grows, the models become a little bit more stable. However, looking at the y-axis here, you can also see that the maximum value on the scale is 0.4. And looking at the full model, the stability doesn't get higher than 0.2, which is really low on the level between 0 and 1. So we found that the overall stability of these models was really poor, as confirmed by this estimator. However, when doing this work, we realized this is a little bit unintuitive. What does this estimator actually say? So the figure I like much more actually is this next one. And for this, we did the same thing, but we looked at the overlap in the number in the top variables. So taking a pairwise comparison between each of those 50 models, take model A and take model B, look at the top 10 predictors, see what is the overlap in these top 10. And in this graph, you can actually see that different target outcome or databases lead to very different risk factors. So across all the grids, you see that the mean overlap is somewhere maybe around 3. Every bar here in the figure represents a different prediction task. And so you can see that for some of the prediction tasks, like here at the bottom, the overlap is slightly higher. So here it is around 8, which is quite high compared to the rest of the results. But overall, when we look across the different phenotype definitions that were used, we can see that the numbers are quite low. So that means even in the top predictors, the ones that we're actually looking at when we're interpreting a prediction model, the differences are huge. And so it's very important if you're going to look at model 1 or model 2 if you're trying to identify risk factors. So the second, actually the third point that we looked at here was what the effect was on the sign of coefficients. And now this matters a little bit less. This matters less for the detection of risk factors, but it matters a lot for the interpretation of them. And so we looked at the sign of the coefficients. And to explain you what you can see here in the graph is there's a count of how often each variable was included in one of the models. And then every variable has like a bar here. And every bar that has one color means that it is consistent across the models in terms of the sign. But any bar that has 2 colors in them, so both red and green, means that for some models, the predicted risk, the effect of the variable on the outcome was positive, and for some, it was negative. Summarizing this in a number, we found that across the different prediction tasks, 46 to 73% of the variables change the sign. Now, there's different ways to look at this, and like from a machine learning perspective, you can explain this. From a clinical point of view, it makes it difficult if you're presenting a prediction model, and it really makes it important to explain that a prediction model is not causal. It really only infers patterns, and it's good at predicting a risk. The risk might be similar, but it might be predicted in different ways. So let me focus now on the implications that it has for risk factor analysis, which is really be careful not to overinterpret these models, because they depend a lot on the study design choices. And actually, not only on the study design choices, but there is also just the inherent randomness in these algorithms that is also still there. So the impact is that there's not a complete and consistent set of risk factors when you use this data-driven approach. And I'm going to let that sink in a little bit. So it is not that the factors that you identify with the model are wrong. The factors that you have identified with the model might just be a very, very tiny subset of the risk factors that there are there for that specific prediction task. They're not generalizable to the whole setting. They're just tied to that specific prediction model, and they might differ if you differ the seeds, you run the algorithm again, or if you choose a slightly different phenotype definition. So why does this happen? The goal of prediction models is focusing on risk prediction. We just want to predict as accurately as possible. using variables might have been different in different models, and also coefficients should always be interpreted relative to the other variables in the model. So it makes sense that these effect estimates change, that the signs flip, and that is correct. But we have to think about how we deal with that when we present those results and how we interpret that. So the goal of risk factor analysis is much more focused on the variables used by the model. And so this is a very important difference that I want you to take away from this half hour. So what do we then do? Risk factor analysis based on characterization is a very powerful starting point to identify a complete and consistent set of risk factors. So we can compare the characteristics at baseline of a certain group of people with and without the outcome. Now this might sound overly simple, but it's just a very powerful and strong way to do this type of analysis. But you don't have to be afraid that rerunning the analysis would give you a different set. So I would argue we have to look at the absolute standardized mean difference to assess the relevance of the differences between these two groups of people. And the great news is that once more, there is OHDSI tools available to do this. So actually several of the existing tools do allow you to compare this group of patients with and without the outcome and would allow you to interpret this for risk factor analysis. So you can, for example, look at the characterization package or the drug utilization package. And the powerful thing is that they're both based on the feature extraction package. And for those unfamiliar with the feature extraction package, it just at a large scale extracts features from all the domains that are included in the OMOP common data model. So both conditions, drugs, procedures, measurements, and you can self-select which ones you're interested in. So I think for risk factor analysis, you would want to have all of them included to be complete. So then to give a little bit of an example, say we're interested to identify risk factors for people using ACE inhibitors to experience an acute myocardial infarction. We would look at the question, which baseline characteristics differ among this group of patients for people with and without the outcome. And for this, I used evidence that was accumulated in this Shiny application, and you can explore it later yourself. I gave you a little bit of a roadmap to find the results I'm going to show you now, because it took me a couple of times to find the table. But it allows you to look at all the factors that have been included in this analysis and to see, you see here the columns, you can see what the percentage of cases in people with and without the outcome, I think. Yeah, with and without the outcome. And you can see the absolute standardized mean difference here. So you just have to make sure you order that correctly, and then you can look at the top variables there. So then you get the biggest difference that there is between the two groups, and those you can go through one by one to see if you recognize them. Are those risk factors that you're familiar with, are they unknown? If they're unknown, can you explain why they're included in here? Is there anything off? Or is this something that requires further investigation? Yes?
So if you run like the default 100,000 covariates from feature extraction, you'll get a list back of 100,000.
Yes, yeah.
How far down do you go?
Good question. So the question here is, if you use all the options and the most elaborate settings in feature extraction, how far do you go down? This is why you definitely order, right? I think it really depends on what you're interested in to do. I think the best way would be to develop, we don't have that, but to be honest, the best way would be to develop a systematic approach to then, after we have identified potential risk factors, to interpret and contextualize those. So I think right now it would be dependent on what you're interested in to study. So what is the reason you're looking at it?
I think just to add to that, what we most often see as like a use case is, so you have that overwhelming table of 100,000 things. So what are you going to do? So as Aniek said, you could just sort it and say, find the biggest amongst the 100,000. That's a very common use case. What that will do is find the biggest absolute standardized mean difference. However, it might be that you're like, well, what I really want is amongst those that are prevalent, what is the biggest difference? So then you may want to filter the table based on those proportion counts. The other thing that I see happen, like in my team very often, is someone asks the question, is diabetes a risk factor? In which case you actually want to search the table. So just to like double down on what Aniek's saying, like when you are complete and consistent, you have everything and you actually can explore the data in multiple different angles to get what you're looking for.
Can I also add one point? So when you see it right now, it's also on the concept level. So it's showing you all the covariates on the code level. If you can also be targeted and feature extraction allow you to do that, and those tools allow you, you can define definitions, cohorts that you as a priori know you're interested in and you want to see it on the cohort level, and you can add that into that mix and you can see it in the table as well.
Yeah, thanks. Good question. Yeah. So then to finish off almost, how to combine the different tools for risk factor analysis. This will just be a summary of everything that I've been through, but I think it's good to have this overview. So the characterization package is a very useful resource to identify all the potential risk factors. So it's very good at the identification part. However, it might not be an accurate depiction of the strength of this relation, because you purely look at a univariate analysis between the risk factor and the outcome. There might be other variables that are also affecting that relationship, and so the effect might be due to something else that is either a collider, a moderator, any of the variables I showed you earlier. So characterization package to identify. Then the cohort method package can be used to estimate a reliable strength between the different exposure outcome pairs, because then you correct for the effects. However, this is computationally more expensive, and so therefore I think it's useful to do first this large exploratory analysis of the risk factors you're interested in. And then finally, the patient level prediction package, although I already said earlier you should not use it, reality might be that you are actually developing a prediction model for another reason. And so it is not wrong to interpret those as risk factors. Those could be risk factors. It's just important to realize here that it is a subset of the potential risk factors. So that is what is the takeaway for the patient level prediction package. And then specifically, this subset might be less reliable when you use more model regularization. So for example, in LASSO, you're also penalizing variables to not be included in your model. So those might be additional effects that might cause this subset to differ even more. And if you want to look at prediction models and use that for some kind of risk factor analysis, then what you really need to do is look at a whole group of models. So for example, the 50 that I presented, you could also maybe explore all the variables that are included in all these models and consider those as the risk factor set. Or we usually, when we're training a prediction model, we're ending up with one model, but actually there might be other models that are almost as well performing, but maybe with a 0.0001 difference in the loss function or so. Maybe look at the class of the 5% best performing models. All these things could at least help you to identify a more complete set. But so remember, for the patient level prediction and for prediction in general, it is a subset and therefore not complete and consistent. So then to end with the takeaways, the risk factor is any characteristic, exposure, or condition associated with an increased risk of developing a disease or an outcome, not causal. It can be very interesting for several reasons, including the discovery of new risk factors for early intervention and personalized care, and how we just answered that in a couple of different ways, but really the way to go is just to characterize the group with and without the outcome to identify the whole set of risk factors, and then do follow-up research to investigate the strength. So if there's any questions, let me know, and otherwise I'm going to give the floor to Azza, I think.
So what I'm gonna do, we have less than 30 minutes left to gather, and we have this demo that I wanted to walk you through, but we also have the closing from Patrick. And yeah, so let me first go on the slides. So I'm gonna try to focus on the most important things that might be important for you today from the demo side of things. So you get some time with Patrick, because I know I'm great, but I know you want to hear from Patrick too. He doesn't think so, but okay. So so far, you learned about how we can phrase our clinical research interest that sits under the bucket of characterization into a formalized research question that is standardized. You saw many different examples of OHDSI community implementing clinical questions in the characterization bucket. And you saw that we have multiple tools and packages to do that, both in OHDSI and in Darwin. What I'm gonna walk you through right now is, in OHDSI, we wanna generate evidence, and we wanna also visualize these evidence, put this evidence in a way that can make sense to our the people who's gonna use the evidence. So we do this for characterization, we do this for estimation, we do this for prediction. So we have an applications that we put out that ran after you run an OHDSI tool, for example, the incidence rate package or the characterization package in Strategist that Sydney went through. You can see those results and we can share it in the public domain. So that's what I'm gonna walk you through. To simplify this exercise, we're gonna pretend that one of the questions you came up with in the previous exercise was something related to GI bleed and something related to the use of celecoxib as a drug. And for example, somebody might have asked the question, and again, the standardized format, saying, among patients with GI bleed, what are the higher demographics? What are their demographics? What the age and gender, prior conditions, medication, and health service utilization behavior. So it's a natural history of GI bleed that we want to explore. You would run it in one of the OHDSI, the characterization package in Strategist that Sydney showed us some of, and now you have the results and you want to explore them. You want to see how you explore them. You can also have a question, like on the incidence rate things. For example, among patients who are new user of celecoxib, how many patients experience GI bleed while on treatment? So that's an incidence rate question. And then you have many others, but for time, I'm gonna try to show you where would you find those answers. You can walk along with me. It's on the results.ohdsi.org, which is a public site that you can see these results. And you'll find it right here. So if you type results.ohdsi.org, you'll see multiple different OHDSI studies that were published on the public domain. If you go into this particular study, which is OHDSI Analysis Viewer App Demo, you will find this particular viewer of the results. So here you see multiple, as we said, we think of analysis in OHDSI that sits under characterization, prediction, or estimation buckets. And today we talk about characterization. So you will see your characterization results in the characterization tab. Okay, if you remember anything out of today's tutorial, you'll remember that we have clinical questions. We're gonna formalize them in a standardized way. That will give us input on target, outcome, time, and risk when we talk about characterization, right? So if you remember, my question was, among people with GI bleed, what's their natural history? So what would be the target in that case? What's the target population? The question is, among people with GI bleed, what's the natural history of that? Exactly. So the app will be exactly customized in that way. So the first thing for the characterization is that you're going to select your target. This particular result is for the questions that I have listed. So you will see here celecoxib, because there was a question about celecoxib, and you will see GI bleed, and you'll see some other drug. So you're gonna pick GI bleed. The reason why the app shows you target group and target is because as Sydney showed you, when you run strategist or a characterization package, you can identify nesting. So you can say, I want people with GI bleed who are pediatrics, so you have this kind of groups, target groups within a bigger target. So the app also allows you to do selection. It also allows you to nest within people who has a 1 year of a prior observation in the data. All these different specification that you might put on your own target population. So you'll pick your target and you will select it. Okay. So the first question we're going to answer is the natural history question. And you will see multiple tabs here in front of you. Because I'm interested in the natural history, I only have one target population, so ignore that first set and look at the second set. And if you're in OHDSI, you know that we don't ever generate evidence from one data source, or at least we don't think of this as the best way of generating evidence. We think that we always need network studies or data or evidence generated from different data sources. So you can always see the results in comparison between different data sources. For example, Cynthia had a question about the treatment pathway among pediatric patients with epilepsy, and she was interested in seeing how those treatments differ by country or by different data sources coming from different countries. So you might want to see the data stratified by different data sources. So you will say database comparison. For this exercise, I have ran the analysis, or the analysis was run on 3 different data sources that all have been ETL to OMAP. So you have data from Germany, you have data from France, and you have data from Medicaid claims data in the US. So you'll pick all the data sources and you will hit generate. That will take you into a table. Now, it's a characterization. It's natural history, right? So you're technically seeing the entire set of variables for patients that met the criteria of GI bleed, depending on your phenotype definition. And you're saying these variables in different tables. The app classify 3 different type of variables. So you have count, binary, and continuous. They binary are variables that are classified, like yes, no, or classifiers, and continuous are continuous variables. Okay, so the first thing you want to know are the counts, because you want to know what is your denominators here. How many people met that target population? So you're characterizing patient based on what sample size. So you'll see here that we have a few thousands in Germany, in France, 100,000 in Germany, and you have 700,000 plus in Medicaid. So this is your counts. This is the sample size for that characterization. And you'll see that in counts. Now, you'll see in binary, you'll see the full set of variables. It will take a look. To answer the question, if you're not looking at risk factors, you're looking at all GI bleed, and you look at all the binary variables that exist in the data model, all the variables, you're here having more than 650 pages of data, because that's characterizing every single concept exists in the data and showing you the variation, the distribution of those variables among people with GI bleed in 3 different data sources. You see the count and you see the proportion. So if you're interested in gender, I can tell you before I put this, any intuition of how will the gender distribution going to be among people with GI? More female, more male, similar. Raise your hand if you think it's going to be a similar distribution of female, male. Raise your hand if you think it's more female. Raise your hand if you think it's more male. Okay, so I see you guys think it's more male. At least that was let's see. So we can type gender. And let's look at the proportions. So in Medicaid, there were way more females. 61% of the people GI bleed were actually females. That might be related to maternity stuff. And then 38% were males. In France, in Germany, we see a little bit of a more balanced with slightly higher in males. And in France, you see slightly higher in males. So very different story depending on the data source. And expect this all the time whenever you do network studies. Okay, similarly, you can look at age. Here, age, we're looking at it as not as a binary variable, or not binary, the right term, as a, oh my God, not continuous, as a categorical variable. And it's 10 age groups, so they're going to be long. There are going to be many of them. I'll show you them here. And it's not nice that you can't really sort them, because it's once you sort, it's alphabetical. So the 0-4 doesn't sit under the 5-10. But you can see that you are seeing GI bleeds in all different age stratas. You'll see that they tend to be higher among higher ages. So you'll see it start kicking in. For example, in France, you'll see the proportions getting higher once you go over 50. Or actually, yeah, over 40. But you see the trend with age with this. You can also see the age as a continuous variable. And you can here you can sort by any data source you want. You'll see 100% for all conditions that are actually GI bleed. Remember, here I'm looking at natural history. So the time frame that I'm looking at those distribution of the variables is at baseline. So at the time people have GI, and I'm looking backwards either 30 days or a whole year. So because it's a natural history question, I'm not interested of the distribution after you have a GI bleed. I'm looking at the time a person has GI bleed or the year before, so quote baseline. Okay. In OHDSI, also common data model, we actually have variables that are interesting in terms of comorbidity, already part of the ETL. So you'll find all the comorbidity index as well. So you will see Charlson index, CHAD, diabetes comorbidity, those all come straight as part of the features. And so you will see that you can see the average of these comorbidity, the mean or the median of these different comorbidity index on the people on GI, if you want to characterize how sick these people tend to be when they have a GI bleed. That's another way that we also characterize standardized right out of the tools. So you see all the covariates, you see those comorbidity index, and of course the counts. And that answers the question, what are the demographics? What's the age? What's the comorbidities? What's the drug that people tend to be on at baseline when they have GI bleed? So I'm going to just quickly show you a bunch of conditions here. So if I filter in condition, I'll be able to see condition, and let me sort by Medicaid. You'll see bleeding going to be 100% because by design all of these people have GI bleed, but here you'll see pain as the most prevalent condition observed in Medicaid with GI bleed. Of course, that makes sense. But then you can characterize all the different conditions that co-occur at the time with GI or at the baseline. Make sense? I still have you? Okay. A second question that our imaginary friend here asked was, what's the, now I know a little bit about GI. I want to know what's the risk of GI bleed among people taking celecoxib. So that's an incidence rate question, similar to what Sydney walked us through. In this case, I'm going to go up. I still want, now, what's my target? Can I keep target as GI bleed? I am looking at among people taking new user of celecoxib, what's the proportion that go into having GI bleed? So my target is new users of celecoxib. So I'm going to pick here, new user of celecoxib. So you see here, I didn't only take new user of celecoxib, I also took the people who have 1 year of observation at the time they take celecoxib because I'm interested with new users. And one way of assert that they're new users is that you capture them in the data, you follow up them for a whole year where they don't have the drug and you see the drug, and then you can say, well, those are new users. So I'm going to select that as a target population. I'm going to do select. And now, very makes sense, I am interested in a incidence, right? Because I'm looking at the incidence of GI bleed. So I click on cohort incidence. I'm going to pick my outcome. In this case, I only have in this app only GI bleed. If you have multiple outcome, you'll pick your outcome. I want to look at all my data sources. As Cindy already told you, by default, incidence rate in packages will stratify by age, by gender, by year. So you can look at things stratified, or for simplicity now, I'm going to look at any. So I'm going to pick all age groups. I'm going to keep the female male because we saw the distribution. Let's actually see how is the risk difference between the two age groups. The observed, we're not adjusting for anything here. We're just looking at the observed. And I'm going to look at all years of data. And the time of treatment that was specified in the package was on treatment here. So I only have one time at risk to select on. When you specify your package, you can have multiple time at risk. You can say, I want to look at while on treatment. I want to look at 30 days after, and so forth. So we're going to hit generate. Oh, my God. OK, that's the last one. OK, so here is the incidence rate. First, you can look at it as a plot. So let's look at it as a plot because that's nice. Here's a plot. Because I select different genders, I actually can look at the gender. So the color is the data source. The y-axis is the incidence rate per 100,000. The x-axis is basically the years, if you had years. So what you see, so each dot is a different data source. A triangle is a male, and a circle is a female. First of all, you'll see how the incidence, the risk of GI bleed among people taking celecoxib in Medicaid is over 2 per 100,000. So it's way higher than the other two data sources. You'll see it's less than 1 in Germany and France. And in all cases, you'll see the triangle higher than the circle. So you'll see the risk of GI bleed is higher in female in all 3 data sources. And you'll see that Medicaid has a much higher observed rate than the other European data sources. So that answered the question, what's the incidence per data source per gender? Now you can also look at it in terms of a table, and you can actually have the exact number. So for example, in France, among the entire database, the risk of GI bleed was 0.13 per 100,000. 0.17 per 100,000 patients, and that's about 13% of the. Hold on, am I looking at this correct? 0.130. Yeah, that's right. Per 100 patient. Yeah, so it's 0.1%, and it's 0.13 per 100 person year. And you'll see in Medicaid it's much higher. We observe 6.6% of the people in the database actually had a GI bleed, so 6 in 100. And that's 2.18 per 100,000, per 100 patients, per 100 person year. Okay, so that answered the question. What's the risk? How it's different in data sources, how it's different by gender. You can stratify by other things, but answer the question of the incidence rate. In the same app, you can answer all the other questions that we talked about today. You saw the risk factor, and you can also find it here. And then you can also see the time to the event. When did the GI bleed happen in relation to the taking celecoxib? You want to hear me or Patrick? Okay, I'll show you time to event because it is, for example, Rima here had an interest in that. So in that case, target is still celecoxib and outcome is still GI, but I'm going to go to no, hold on. Yeah, and I'm going to select. So once you selected exposed summary, it's going to give you the risk factor. That's the tab that that you saw before. You're going to say case series, which you were interested in because it's going to give you the whole characterization of the people who had the surgery. So you'll find this under case series, and it's characterization of the people with the outcome. And you will see the time to event. I'm going to click on it. It's one of the most complicated and hard plots to look at, but I apologize. But it's very, very interesting. Okay. So what you see on the y-axis is the number of events, and you'll see multiple data sources. Yeah. Okay. Okay. Here we go. So you'll see Medicaid. The y-axis is the number of events, number of GI bleed. 0 is the y-axis is time. 0 is the time that people took celecoxib, so that's the index of the target cohort, so that's the time initiating the treatment. Every bar is the count of the event of GI bleed. You have 3 different plots because we're measuring time at different intervals. So in the first one, you're looking at day by day. And you'll see that if you go 1 day, for example, before celecoxib, that there were that many events of GI bleed in that target cohort. So you'll see that GI bleed is happening. Okay, and then you have orange and green, orange and green. These are the events that happened before the target starts, before celecoxib starts. And the blue one are the ones that happened after celecoxib starts. So you will see, if you want to interpret that GI bleed tend to happen the whole time. People get celecoxib, it's still happening after. You'll see a little peak after celecoxib or the day 0. So maybe people are taking celecoxib for the GI bleed. But what you see here is that the events are a lot of events happening before and events happening after. Here you're seeing the same plot, but the unit of time now is 30 days. So you're able to kind of zoom out and see it on in a bigger picture. And you say that you can go all the way back to 2,000 days. You see the same trend. Here you see the histogram, and the time now is year. So every bar is 1 year. And you'll see that there's all these events happening. Here's the start date of celecoxib, and there's more events happening after. So that tells you, and of course, if you're interested, if your research question, you don't care about what's happening in the past, you just want to know when the GI bleed happened for people after celecoxib. So you'll see that it's happening all around. There's a distribution of the events. So there's a bunch of events that happen right after, and then it goes down. You have 5 minutes for Patrick. Oh.
Thank you, Azza. Any questions? Or questions for Azza? Last minute efforts to get candy. So I am going to get you out on time. That's probably the most important thing. But what I do want to just conclude with very, very briefly is a couple things. Oops. Let's make sure that goes out there. Yeah, okay. So I'm just going to highlight just a very couple things. You have now learned about characterization, all the different types of characterization analyses that are available through the OHDSI community. I hope that you take away from this that there exist best practices for how to think about characterization. There exists open source tools for executing, and there exists direct clinical applications of why doing characterizations across the OHDSI network would actually have some demonstrated value. Last year at the OHDSI Symposium, we held a tutorial, and actually Andrew Williams led a tutorial on how to run a network study. Andrew passed away this year, and I wanted to pay tribute to the fact that he did an amazing job setting up this tutorial for our community, and it still has tremendous enduring value to look at. But if you are somebody who wants to think about how to learn a network study, I encourage you to check out this tutorial that's available on our website that was done last year. It was really well done. We have given lots of tutorials on how to design and execute studies that are all available. And actually, Craig Saxon's done a great job of creating a new website that's just called ohdsi.org/tutorials. It's like all the different things that you could learn. This tutorial will be posted on this website, but also lots of tutorials are before. But probably the most important thing I want you to take away from here is if you are interested in designing and executing a characterization study across the community, the main thing to recognize is that that's not a task you need to be doing on your own. The whole point of why we have established this community is so that we can work together and collaborate towards answering important questions that patients deserve the answers to by taking advantage of this tremendous data resource that we've created and take advantage of the collective expertise of our community. And so I want to encourage you to hopefully, as you take something away from this course, maybe you've learned a little bit about characterization, maybe it's inspired you to think about a study, but hopefully more than anything else, it's going to motivate you to want to collaborate with me, with our faculty, and with everybody else in the community so that you can think about how to translate these ideas into actions and make those actions into evidence that the world actually needs to improve public health. So with that, I'm going to actually just stop and thank you all for attending and sticking with us. I'll give you just a quick logistics rundown. If you are a poster presenter, you now have 1 hour to get your poster hung up. And you also have that 1 hour to make sure that you find your little stamp that you were given at the front door, because you're going to need it. For all of you, whether you're a poster presenter or not, our network reception is at 6 p.m. just out in the foyer, and it'll be from 6 to 8. And it is not just any normal network reception. We're going to have a little bit of fun with some fun and games, opportunities to win prizes that are all focused on making sure that you learn from each other in our community and our collaborator showcase. So I look forward to seeing you all at 6 o'clock. And if you have a poster, please do go hang it up because people are going to be coming in to check it out throughout the evening. Thank you all for attending our tutorial. I really appreciate you participating and looking forward to collaborating with all of you. Thank you.